you. We are having a marathon web series starting from today. Today is day 1. Welcome to Challenges in Hematology, which would also include the best of EHA 2022, 27th Congress of European Hematology Association, under the aegis of Mumbai Hematology Group. We can't be thankful to him. We just are so grateful and indebted to our program director, Professor Dr. Niemeyer, sir, for bringing this up for all of us. You're an inspiration, sir. The country is fortunate to have you. I was having a debate with you today, and in last 2.5 years, the quality of hematology care, because of this wonderful web series which you are bringing in under the aegis of Mumbai Hematology Group across the country, has really grown. So a lot of clinicians today are giving the benefit of the best of the treatment across the country, whether it is tier 3, tier 4 towns. No wonder, sir, that you have been awarded the Inspiring Hematologist of India by Economic Times. A warm welcome to all the 3 guest speakers today, and the discussions, and not least, just to mention all the participants from Kenya, Nepal, Bangladesh, UK, Philippines, Myanmar, Sri Lanka, Malaysia, USA, Indonesia, and many more countries, around 25 of them, will be logging in today. A warm welcome to each one of you. Please do not forget, you have to log in every day at 6:25 till 9:30, Monday till next Sunday. Thank you, and may I request our program director, Professor Dr. Niemeyer, sir, to take it from here.
Thank you. Good evening to one and all. Thanks to Dr. Manoj for his kind words. This web series is brought to you by Mumbai Hematology Group. It is supported by INTAS through and through, all the lectures, all the days, the whole series, and managed by Perfect Square. I thank Dr. Manoj Kumar and their team from INTAS for becoming the sole sponsor of this 7 days long 21 lecture series. Mr. Yash, Mr. Kalpesh and the team from Perfect Square for doing all the background work as well as the work for next 7 days. The executive committee of Mumbai Hematology Group. All our discussants were themselves eminent hematologists, medical oncologists, and almost 170 of you have offered to be the discussants for one or the other lecture. So some of you will be there for each lecture depending upon your interest and specialty, and that will make the question and answer session very attractive. And of course, the participants, as Dr. Manoj said, from large number of countries, from any part of the world, who have been interested and who have logged in. Our website is www.mhindia.com. All the information right up to 3rd September for all the academic activity has been put up. The website has been updated today. We update it every week. You can just visit it for what is there to come, and you can probably choose and organize your calendar upon it. So once again, as Dr. Manoj mentioned, Monday to Sunday, 4th to 10th of July, 3 lectures a day. This is going to be a very interesting series. And today is day 1. And we have our first speaker, and it's my pleasure to introduce her, Professor Samira Lakhal-Littleton. She's Associate Professor of Cell Physiology and Tutorial Fellow, MRC Senior Non-Clinical Research Fellow, Department of Physiology, Anatomy and Genetics at Oxford University. She's joined. Yes. Welcome, Dr. Lakhal.
Thank you very much indeed for this introduction. Can I just get some nods that you can see me and hear me?
No, at the moment, yeah, now we can see you. We can hear you.
Brilliant. Well, thank you very much indeed for inviting me to speak. If I may, I'd like to share my screen with you.
Sure.
Can I just confirm that you can all see my screen?
We can see your screen, but it's not in the full slideshow mode.
Okay. What can you see on my screen at the moment?
We can see the main slide as well as the next slide.
Ah, okay. So what I'll do. How's that?
Perfect.
Excellent. Well, as I said, thank you very much indeed for inviting me to speak. So I run a research group into iron homeostasis. My research is based at the University of Oxford. And you will all know that for a long time, our interest in iron has really centered around the erythron. My research and my interests are very much based on or centered around what iron does outside the red blood cell. And so today I will be talking about some of the work we've done, my team and I have done over the past 8 years or so, where we have tried to use a combination of mouse models and human studies to understand the consequences of iron deficiency in tissues other than the erythron. Before I start, I thought I'll give you a little bit of the long view about iron. As I said earlier, we often think about hemoglobin when we think about iron, but obviously iron has been important for biology or in biology well before hemoglobin ever existed. So you will know that it is universally acknowledged that Earth evolved from an oxygen-free environment to a fully oxygenated biosphere. There's been 2 major oxygenation events occurring 2.4 billion years ago and another one 540 million years ago. And obviously, before these events, iron was freely bioavailable in the ocean. However, the advent of oxygen and its subsequent oxidation caused the precipitation of iron, and this process is called banded iron formation. The formation of banded iron actually coincided with the explosion of biomass, leading to increased need for redox active metals that could operate within a wide range of redox potentials. So essentially, throughout the history of Earth's evolution, what we have is a major reduction in the bioavailability of soluble iron, coinciding with an exponential increase in the demand for iron. Bearing that in mind, it is perhaps not surprising that iron plays fundamental role in important biological processes, not just in the transport of oxygen. And so, why does it do that? Why is iron so important? Well, you will probably know that the ferric ferrous iron is a very special redox pair. And it is a special redox pair because the redox potential is modulated by the complexing ligand. And so whether it be a protein or carbohydrates, and that makes it suitable for catalyzing a very diverse range of biochemical reactions across a wide range of pHs. So this is just a sort of a small list of the enzymes and the proteins that use this redox pair for their activity. Unfortunately, we've obviously tended to focus on just one of them, hemoglobin, and we've tended to perhaps overlook all the other important functions that this redox pair carries out. So why do we know that iron is important? What is the evidence that iron is important outside the erythron? Is there any physiological evidence? And the answer to that is there is plenty. I'll give you a few examples of the evidence, the direct evidence for the role of iron outside the erythron. So this is a study looking at pulmonary arterial pressure in healthy volunteers and non-anemic iron deficient individuals. The healthy volunteers are in dark grey, and the non-anemic iron deficient individuals are in the light grey bars. And as you will know, a basic physiological response to hypoxia is to vasoconstrict the pulmonary arteries, and that results in a rise in pulmonary arterial pressure. You'll notice that the non-anemic iron deficient individuals have a heightened response to hypoxia. And if you were to then bring the same individuals a week later and give them an intravenous iron infusion prior to exposing them to hypoxia, you will see that this intravenous iron infusion has removed the difference between the two groups, and furthermore has caused inhibition of this physiological response to hypoxia. Please note the time course of this effect. This effect occurs within 2 to 3 hours before any change in hemoglobin is observed. So this is evidence that both non-anemic iron deficiency and exogenous iron supplementation can directly regulate the pulmonary response to hypoxia independently of hemoglobin levels. Another line of evidence, if we look at individuals with idiopathic pulmonary arterial hypertension, for instance, and we look at the soluble transferrin receptor markers in the plasma, you will all know that this is considered to be a marker of unmet iron need in tissues. You will see that those individuals with soluble transferrin receptor higher than 28 nanomole per liter have worse survival. Soluble transferrin receptor levels are inversely related with iron demand. So these individuals here in green are more iron deficient than those. If you look at individuals with chronic heart failure, you look at the survival over time, you will also notice that while iron deficient anemic individuals do very badly, iron deficient individuals without anemia also do badly. And so again, pointing to perhaps the effect of iron deficiency on heart function independently of anemia. And these are data from our own work looking at maternal iron deficiency and its association with peripartum cardiomyopathy. And similarly, you can see here that there is a strong association between low soluble transferrin receptor levels and the risk of peripartum cardiomyopathy. So as I said, the evidence that non-anemic iron deficiency can impact or have an impact on our physiology has been around for a long time. The dilemma is how do you explore the function of iron outside the erythron without the confounding effects of anemia? Typically, people might look at animal models of dietary iron deficiency, but those models also develop anemia. In clinical settings, it's extremely difficult to find or define a group of individuals who are iron deficient only and not yet anemic. And so looking or trying to explore the function of iron outside the erythron is not straightforward. And I'll explain why that is. So I'll give you a little introduction to systemic iron homeostasis. So many of you will already be familiar with this, but essentially we rely on 3 sources of iron in our body. The primary source of iron is the recycling of red blood cells in the spleen. We get some iron daily from the duodenum, from dietary absorption. And when required, we also release some iron from the liver stores. For iron to be released from these 3 sources, we require the iron exporting protein ferroportin. In the plasma or the blood, iron is carried by its chaperone transferrin. Most of it is used in the bone marrow to generate more red blood cells, but some of it also is taken up by tissues. You will also know that the process of systemic iron regulation is dependent on the liver-derived hormone hepcidin. Hepcidin is upregulated in response to raised transferrin saturation, and it acts to inhibit ferroportin function and restore normal transferrin saturation in the blood. So how about in tissues? How are tissue iron levels regulated? So let's talk about cellular iron homeostasis. So you will all know again that iron homeostasis in cells is controlled at the site of uptake through transferrin receptor, storage in ferritin, and when cells are iron deficient, they respond by increasing the uptake of iron by increasing expression of transferrin receptor and decreasing the expression of the ferritin protein, stored protein ferritin through these iron stores. Conversely, when cells experience excess iron, they will reduce the expression of transferrin receptor on the surface and increase the expression of the iron storage protein ferritin to ensure that iron levels inside the cells remain below the levels at which reactive oxygen species are generated. However, work over the past few years in my own team has discovered an additional mechanism of cellular iron homeostasis. And I should also add that this additional mechanism of cellular iron homeostasis is non-redundant. I'll explain that a bit later. So we've discovered that some cells, not all, some cells also utilize ferroportin, the iron exporting ferroportin, not for systemic iron homeostasis, but in this case for the control of cellular iron levels. And furthermore, some cells also produce their own hepcidin so that they can regulate their iron export in a cell-autonomous manner. So it appears that some cells in the body utilize an additional mechanism of cellular iron homeostasis, which is regulated export. Now, when we made this discovery, it gave us an idea where we thought, well, if we were able to specifically target this local hepcidin ferroportin axis, we may be able to induce tissue iron deficiency in a localized and targeted way without causing anemia. And that would help us to try to understand the consequences of iron deficiency in these tissues without affecting hemoglobin levels. So that's what we did. And in the space of sort of 7 or 8 years, we were able to do that in a number of tissues and organs, including the cardiac myocytes, pulmonary arterial muscle cells, fetal hepatocytes, and renal tubules. So how did we do this? We generated 3 mouse models, each enabling the targeting of either ferroportin, hepcidin, or the knocking of a hepcidin-resistant ferroportin. So this hepcidin-resistant ferroportin replaces the endogenous ferroportin, and this ferroportin variant exports iron to the same extent as the wild-type ferroportin, but it cannot be inhibited by hepcidin. And we crossed these models with a tissue-targeting creocombinase mouse, where the creocombinase that targets these sites, these LOX-T sites, is expressed either the renal tubules, cardiac myocytes, liver, or in the smooth muscle cells. And then we did some painstaking work over a number of years to try to understand the consequences of disrupting local regulated iron export in each of these organs. In the heart, the first thing we did was to generate cardiac-specific ferroportin knockouts, i.e. mice, where cardiac myocytes are unable to export iron. These mice systemically are normal. Their hemoglobin and plasma iron levels are normal. This shows that ferroportin in the heart is not involved in systemic iron homeostasis. However, what we do note in these mice is that they develop fatal heart failure as a consequence of cardiac iron toxicity. we're able to prevent heart failure in these mice by putting them on a low iron diet. So you can, to some extent, mitigate against the effects of inhibited export by reducing uptake. Now, when we generated mice lacking hepcidin in cardiac myocytes, those are cardiac myocytes that are leaky for iron and cannot retain iron. What we found is that these mice developed heart failure as a consequence of cardiomyocyte iron deficiency. The hearts in these mice shifted to glycolysis as a primary mechanism of ATP generation, as opposed to oxidative phosphorylation. And in these mice, we were able to prevent heart failure by giving intravenous iron treatment. Again, suggesting that you can, to some extent, mitigate against the consequences of increased export in the cardiac myocytes by increasing uptake or iron available for uptake. So these data were the first evidence to show that iron deficiency within the myocardium itself, even against the background of normal hemoglobin levels, is sufficient to impair contractile function of the heart. When we generated mice that expressed the hepcidin-resistant ferroportin within the cardiac myocytes, they developed the same phenotype as those lacking cardiac hepcidin, suggesting that cardiac hepcidin operates in a cell-autonomous manner to regulate iron export. And then this regulated iron export within the myocardium is required to maintain adequate iron levels for normal heart function. Now, we used the same strategy to look at the role of iron in the pulmonary vasculature. So what we did was we generated mice that expressed this hepcidin-resistant ferroportin in pulmonary smooth muscle cells. What we found is that these mice developed pulmonary arterial hypertension and consequently right heart failure. What we found was that smooth muscle cells of the pulmonary arteries expressed high levels of ferroportin as a consequence of loss of hepcidin regulation, became iron deficient and behaved as if they were hypoxic, i.e., they increased the expression of hypoxia-regulated genes such as endothelin-1, and we know that endothelin-1 is a vasoconstrictor involved in the development of pulmonary arterial hypertension. Furthermore, we were able to prevent and partially reverse the development of pulmonary arterial hypertension in these mice, either by giving intravenous iron or by inhibiting the signaling endothelin receptor 1 signaling. This was the first line of evidence that iron deficiency within the pulmonary vasculature is sufficient to cause vascular remodeling and drive the development of pulmonary arterial hypertension and consequently heart failure. Now, interestingly, what we found when we looked at pulmonary arterial smooth muscle cells from individuals with genetic or hereditary pulmonary arterial hypertension, these individuals have BMPR2 mutations, bone morphogenic receptor 2 mutations. When we looked at these cells, we found that they phenocopied the cells derived from iron mice, i.e., they expressed very high endothelin levels, and they were also relatively iron deficient when compared to smooth muscle cells from healthy individuals. Interestingly, it appeared that these BMPR2 mutations that cause hereditary pulmonary arterial hypertension appear to cause a decrease in local hepcidin production within these cells and consequently an increase in ferroportin. So it may be that the mechanism of iron regulation in smooth muscle cells that we found to be essential is in fact impaired by BMPR2 mutations and may be a contributing factor to disease in this group of individuals. And likewise, we were able to prevent and partially reverse the development of pulmonary arterial hypertension in these mice. And it would be interesting if the same applies in patients. The role of iron in fetal hepatocytes. This particular line of inquiry was really instigated by the notion that fetal hepatocytes express hepcidin quite early on in development. And we were interested to see, in fact, we were convinced that this fetal hepcidin was going to be responsible for regulating the amount of iron acquired from the mother through the placenta. The hepcidin target ferroportin is very abundantly present in the placenta. And so we thought, we were in fact reasonably confident that it was going to be a matter of fetal liver hepcidin regulating ferroportin in the placenta. That turned out not to be the case at all. When we generated mice that lack hepcidin just in the hepatocytes and looked at the fetuses in the second and third trimester, what we found was that these hepatocytes were unable to build up iron stores. So I should say here that if you look at both the fetal, both the mouse and human placenta and human livers, there is a very rapid exponential increase in the amount of iron deposited in the liver in the third trimester of gestation. And this is the buildup of iron stores required to sustain growth at this stage, but also in early neonatal life. Now, interestingly, it appears that hepcidin is required for the buildup of those stores because the animals that lacked fetal hepcidin, fetal liver hepcidin, were unable to build up adequate iron stores. And you will all know that there is a level of hematopoiesis that occurs in the liver in this late stage of development. And so what we did find was that these fetuses had impaired hepatic hematopoiesis and had transient fetal anemia as a consequence of lack of iron buildup in the liver. So this told us two things. The first is that hepcidin, fetal liver hepcidin, is required for the buildup of stores in the third trimester. And the second information we learned from this is that hepatocyte iron stores in the third trimester of development were required for supporting hematopoiesis. Likewise, when we generated mice that express the hepcidin-resistant ferroportin in fetal hepatocytes, and then we looked at the fetuses in the third trimester, their phenotype very much phenocopied that of mice lacking fetal liver hepcidin, again suggesting that hepcidin operates in a cell-autonomous manner in its context. Now we move on to the kidney. We generated mice that lack ferroportin, specifically in the renal tubules. And what we found was that these mice developed renal iron overload and decreased iron reabsorption. And this confirmed that ferroportin in the renal tubules was involved in iron reabsorption from urine back to the blood. And this resulted in reduced iron in the plasma and transient anemia. These mice recovered from the anemia by increasing iron absorption in the gut. So while it appears that ferroportin in the renal tubules is involved in iron reabsorption and control of iron in the plasma, it did not have an essential role because it could be compensated for by changes in dietary iron absorption. Now, conversely, when we generated mice that have expression of hepcidin-resistant ferroportin, so this is the leaky ferroportin that cannot be inhibited by hepcidin, and this expression is targeted specifically to the renal tubules, what we found was the opposite of the model I described earlier. So we found that these mice developed renal iron deficiency due to excessive iron reabsorption into the blood. And interestingly, these animals were sensitive to iron-rich diet. So they appeared to reabsorb more of the iron back into the plasma, and as a consequence developed greater iron overload in other tissues, but lower iron overload in the kidney. So this body of work, which I said we've carried out over a number of years, has taught us some lessons, but have also given rise to some questions. I think the most surprising finding for us is that iron has appeared to, cellular iron outside the erythron appears to be essential in really fundamental physiological processes. I showed you from the pulmonary arterial hypertension model that iron in smooth muscle cells is required for the control of vascular tone. I told you as well about the mouse model of liver-specific hepcidin knockouts, and we found that the buildup of iron stores in the third trimester of development was required for supporting hematopoiesis. We've shown you how iron levels in the renal tubules were tightly controlled by hepcidin flow 14, and how there is an inverse relationship between tubular reabsorption and systemic iron levels. And in the heart, I showed you how iron deficiency within the cardiac myocytes, again independently of plasma iron levels or of anemia, was able to impair contractile function. The questions that we are now asking ourselves is how? How does iron control vascular tone in smooth muscle cells? How does iron control contraction? The other question we're asking ourselves is where else? Where else is cellular iron key or important to the physiology of the tissue in question? We're starting to get a handle on the how question. So, as I told you earlier, the iron-deficient cardiac myocytes appear to switch away from oxidative phosphorylation as a main mechanism of energy production to what appears to be a pseudo-hypoxic response. This is, in a way, completely expected. It is not surprising at all. You will know that many of the enzymes and complexes in the mitochondria that carry out with our components of the electron transport chain require iron in one form or another, either in the form of heme or iron sulfur clusters. So it is not surprising that cardiomyocyte iron deficiency leads to a reduction in oxidative phosphorylation. The surprise was that these iron deficient cardiac myocytes appeared to switch to glycolysis. This is a response normally seen in response to hypoxia, not in response to iron deficiency. But when you take a look at the oxygen sensing pathway more closely, it all becomes clear. So you will know that oxygen sensing in cells is carried out by the transcription factors, hypoxia-inducible factors HIF 1 and 2, and that these proteins are constantly being synthesized and then broken down by the action of poloyl hydroxylases and FIH, factor inhibiting HIF. Now, this breakdown requires oxygen as a co-substrate and iron as a cofactor. In hypoxia, when oxygen is not present, the co-substrate is missing, these transcription factors are not degraded, they stabilize and they translocate to the nucleus, where they drive the expression of a number of target genes, including glycolytic genes and endothelin-1. And so, because iron is a cofactor required for this degradation of HIF, iron deficiency in the cells will engender a pseudo-hypoxic response and will also lead to the stabilization of HIF and the trigger of a pseudo-hypoxic response. And that explains why the iron-deficient myocardium shifts towards glycolysis, even though it isn't hypoxic as such. Now, why are all these findings important? You will all know that iron deficiency is increasingly being recognized as an important comorbidity in chronic conditions. And this is independently of anemia. And iron deficiency is associated with greater mortality and adverse outcomes in chronic heart failure, chronic kidney disease, cancer, and lung disease. And so we know that this iron deficiency must be operating via mechanisms other than hemoglobin. And we have to consider those mechanisms in our approach to treating this iron deficiency. And I think this slide hopefully sums up what I'm trying to say. So up to now, we have concerned ourselves primarily with the reduction in hemoglobin when looking at individuals with iron deficiency. And of course, anemia will have an impact on these organs through reduced oxygen delivery, no doubt. But what is apparent is that local iron deficiency within those tissues also has a direct effect, independently of anemia. And it appears that there are different mechanisms operating in different tissues in response to iron deficiency. The question that comes to mind first is, when should we intervene with iron supplementation? Should we wait until anemia develops, by which stage these tissues have already experienced local iron deficiency? Or should we perhaps intervene early on to prevent these tissues from becoming iron deficient? And if that's the case, then on what markers should we base this intervention? Currently, we base intervention on hemoglobin values. But if we wanted to target local iron deficiency, we would need preferably a plasma iron marker that tells us that the heart is iron deficient, or the lung smooth muscles of the iron deficient, and so on. We currently don't have such markers. Soluble transferrin receptor has, to some extent, been suggested to be a marker of unmet tissue iron demand, but it isn't specific to any tissue in particular. The other question that comes to mind is, all of the iron supplements that have been developed, the primary outcome in the trials has always been raising hemoglobin levels. We have not been concerning ourselves with correction of iron deficiency in tissues when developing iron supplements. And I showed you earlier from some of the work in mice that some of these supplements do also correct iron deficiency. But the question is, should we design iron compounds that target specific organs? The current supplements that correct hemoglobin levels are, you could compare them to a sledgehammer approach, where you have a large quantity of iron being given and it floods the system with iron. We have no control over where this iron is going. And it may be that we should perhaps consider targeting iron deficiency in specific organs. Or maybe one way of achieving specificity is to target the mechanisms that are evoked by iron deficiency in the individual organs. So I told you earlier, for instance, endothelin 1 being evoked by iron deficiency in the pulmonary arterial smooth muscle cells, glycolysis being evoked by iron deficiency in the heart, and so on. So lots and lots of questions, and as I said, the work that we've done over the past few years has given us some answers, but many more questions that we look forward to exploring over the coming years. With that, I'd like to end by acknowledging all my lab members who have worked on this in one way or another. Dr. Graham Mohammed was involved in the kidney study, and in one way or another, all the other studies. Dr. Mayra Aviles, who's currently working on the mouse model of heart failure. Sinaida Sherubin, who has been working on the collaboration with peripartum cardiomyopathy cohorts. Lara Camara, who worked on the mouse model of fetal hepcidin knockouts, and all the other students who have worked on other aspects of the project. My funding, primarily the Medical Research Council, the British Heart Foundation, and all the collaborators who have worked with me over the years in Oxford and outside. And I'll be happy to take any questions. Thank you for attention.
Thank you so much, Dr. Lakhal. That was wonderful. Very different angle. We haven't heard this kind of talk earlier. Majority of us listening today to you are clinicians, hematologists who practice treatment of patients. I have one question. In the intrauterine life, iron deficiency leads to retarded growth of the brain function. That has been something which we keep talking about. Did you think about looking at that?
Thank you. So there is rich evidence for the effect of iron deficiency, prenatal iron deficiency on brain development, cognitive function, fine motor skills, all those things. The mechanisms have been studied widely by others. It appears the main mechanism that comes up is a failure of myelination. Myelin synthesis heavily requires or is heavily dependent on iron availability. So that's one key mechanism. But we haven't ourselves looked at any other mechanisms.
Right. And when you're talking about the biomarkers, and you did mention about soluble transferrin receptor, so things like total iron binding capacity, soluble transferrin receptor, ferritin, when they are indicating iron deficiency with a normal hemoglobin, are these good markers?
To an extent, they are the best we have now. And I suppose what I'm trying to argue for is perhaps we need to look at slightly better markers that tell us about specific iron demand in particular tissues. And really the only way we can do that is by honing in on individual tissues, understanding the pathways that are activated locally by iron deficiency, so that we can follow on from that and try to identify markers in the blood that will specifically tell us that the heart is iron deficient or the vasculature is iron deficient. We don't have that yet.
Yeah. Now what happens to the immune system in iron deficiency?
Yes, so this is the work that is currently the focus of my colleague Alexander Drake-Smith, also in Oxford. And he's shown that T-cell activation requires iron, is heavily dependent on iron. So both, and this is particularly true in response to vaccines. So it appears that iron deficiency will also impair, again, independently of anemia, will also impair vaccine response.
Right. We have some of our colleagues who have raised hand to ask you a question. Let's start with Samir.
Hi, good evening. From what I understand, so you've had this hepcidin, the intracellular iron is getting lost. It's going out through the ferroportin channels into the blood, and that's why
Samir, we are not able to hear you.
Intracellular iron deficiency is what I understood. So, from what I understand, you know, okay. Am I more audible now?
Yes. Yes, Samir, you're audible. Repeat.
Yeah. So, from what I understood, there were you had hepcidin knockouts. Wherein which, because of loss of hepcidin, the intracellular iron is not retained and it is expelled from the cells, and that's why there is a tissue iron deficiency. So is there actually a situation in real life where there is where patient deficiency? And secondly, anemia would develop much earlier generally as compared to so iron is retained for all these essential cellular mechanisms, like for example cytochrome B, and such other cellular functions. Iron is retained for these functions, and then it's only so would anemia actually develop much earlier before you have impairment of cellular functions?
Okay, thank you. Sophie, you broke up a few times there, so I'll try to answer the questions that I understood. So if I understand correctly, you're asking whether there is a real life situation in patients where we have a leak of iron out of cells. So what we do have is individuals who are iron deficient, who have iron deficiency anemia. So individuals with iron deficiency anemia will have both anemia and will have low hepcidin. So hepcidin has got a very intimate relationship with the bone marrow. The bone marrow, the anemic bone marrow, will actively suppress hepcidin. So in individuals with iron deficiency anemia, you will have both the impact of anemia, but also the impact of the hemoglobin of the bone marrow on hepcidin. And the hepcidin will be very, very low indeed. So they will be both deficient in terms of iron supply in the blood, but also their hepcidin will be suppressed and their cells will be leaky. And so these tend to be individuals with iron deficiency anemia or with absolute iron deficiency, which is quite different from individuals with functional iron deficiency who will have typically high hepcidin because of inflammation. So that's your first question. The second question, I think you said whether anemia develops before tissue iron deficiency. All the evidence we have is to the contrary. Tissue iron deficiency develops before anemia. Anemia, in fact, is the last consequence of iron deficiency. The bone marrow is very, very good at holding on to iron and getting all the iron that is in the blood. It has the highest expression of transferrin receptor. It's a very, very, I would say, very absorbent sponge of iron because of the amount of transferrin receptor expressed in the bone marrow. So typically when you have iron, on the way to iron deficiency anemia, other tissues become deficient because their ability to take up iron from the blood is a lot less than that of the bone marrow. Additionally, the bone marrow speaks directly to the liver, as I said earlier. So the moment that the iron deficient, the bone marrow experiences an increase in EPO because of anemia, what we do see is a suppression of hepcidin. So the bone marrow is able to have a say, so to speak, in how much iron is available in the plasma, whereas the heart or the vasculature or the kidney do not necessarily have that say.
Okay, thank you so much. Yes, you did. Thank you.
Thank you, Dr. Samira. There's a question from the audience. What would be the missing link between iron deficient anemia, ferroportin, and vitamin K bleeding during the birth?
And vitamin K?
Yeah.
Okay. That's a really interesting question. It's not something I have thought about. I can tell you that bleeding during birth is another compounding factor in maternal iron deficiency. So maternal iron deficiency is caused both by the amount of iron that the mother deposits into her fetus. So we give a lot of iron stores. We selflessly donate it to our growing fetus. But also during birth, there is a major component of iron loss at that stage. But how vitamin K influences this, I'm not sure.
Something unusual. Okay. Dr. Rajat, your question.
Yeah. Dr. Samira, fantastic talk. My question is about immune system again. You said iron deficiency interferes with the T cell activation. Does it have any effect on neutrophils or macrophage function? Because we know, as a clinician, we avoid iron supplementation in a child with active infection, and we know USNA infection is not uncommon during iron chelation. So does iron have any effect on neutrophil or macrophage function, or is that not being studied?
So the relationship between iron and infection very much depends on the type of the pathogen that you're talking about. So we know that iron supplementation can increase the risk of infection for certain pathogens, while iron deficiency increases the risk of infection for other pathogens, and it very much depends on where the pathogen lives. So for those bacteria, for instance, that do reside in macrophages, we know that iron supplementation will increase the risk of infection and will worsen outcomes. And this is one of the debates, the hot debates in developing countries, whereby, for example, in sub-Saharan Africa, there is this debate about whether we should supplement children with iron in malaria endemic regions because of this requirement of the malaria parasite requiring iron and benefiting from iron. So I think the answer to your question, it really depends on where the pathogen lives.
Okay, thank you so much.
Thank you. One question from the audience. He wants to know your comments about neurodegeneration and brain iron accumulation.
Thank you. So this is a common phenomenon in a number of neurodegenerative disorders where we have focal iron deposits. We see this in Alzheimer's, Parkinson's, and so on. And I think we know that a number of cells, astrocytes, glial cells, neurons themselves express the reporting in the brain. And so you can very well imagine that we know that aging is a disease of an inflammatory disease, and we know that hepcidin goes up in the aged brain, in the aging brain. And so what likely causes this focal iron deposit is the local inflammation in the brain, raising hepcidin and blocking iron export out of these ferroportin expressing cells.
Thank you. Dr. Chandran.
Thank you very much for the nice talk. I have two questions. One, is the term local iron deficiency synonymous with functional iron deficiency, especially in case of anemia of chronic disorders? And second one is, I think I missed the part during your presentation, like what are the threshold points for various biomarkers like ferritin or soluble transferrin receptor levels to intervene in case of this local iron deficiencies?
Thank you. You ask really good questions. So local iron deficiency is not currently defined at the clinical level. It's an entity we have discovered to have an effect in human studies, in controlled human studies and in mice. But at the clinical level, we have no means of defining local iron deficiency. Soluble transferrin receptor, an increase in soluble transferrin receptor has been suggested to be synonymous with unmet tissue iron need, but it isn't specific to a particular tissue. So as I said, one of the main goals moving forward is to try and see if we can identify markers of local iron deficiency. We don't have them at the time, at the moment. Functional iron deficiency is very much one that is driven by inflammation. And you can imagine that inflammation, one of the reasons that individuals become iron deficient is hepcidin goes up. Hepcidin is a direct target of interleukin-6, and hepcidin will block iron export from the gut. So we stop absorbing iron, but we also stop releasing iron from the spleen and from the liver. So actually functional iron deficiency isn't in fact an iron deficiency per se. It is a blockade of iron of iron export. So it is a sequestration or maldistribution of iron. Most of the individuals who have functional iron deficiency, in fact, have normal total body iron stores. The only problem for them is that iron cannot get out of the spleen and cannot get out of the liver. And you can imagine as well that for any any organ that expresses ferroportin, there will also be a blockade of ferroportin in those organs, including the heart. So we think that there will be a very big difference between individuals with functional iron deficiency who have high hepcidin and individuals with absolute iron deficiency who have suppressed hepcidin, because the distribution of iron in different organs between those two groups will vary greatly.
Thank you.
Thank you.
Dr. Samira, I have one more question. The fetal liver hematopoiesis. My understanding was it is maximum in the first few months of pregnancy, first trimester, and it sort of vanishes by the late pregnancy. Is that correct or not?
So what we found was that in the second half of gestation, fetal liver hepcidin appears to be required for the buildup of rapid iron stores. So what we've seen is that there's a rapid buildup of liver iron stores, and this is to give an endowment of iron so that it supports growth in early neonatal life. If we take away hepcidin in this late stage of development, in the second half of gestation, that buildup doesn't happen, and the fetus is reduced iron stores and transient anemia. That anemia corrects quickly because, as the fetus is born, hematopoiesis shifts back to the bone marrow. It no longer depends on the liver.
Thank you. Dr. Abha, your question.
Thank you, sir. Thank you, ma'am, for the very comprehensive lecture. Ma'am, this just got me thinking in diseases like polycythemia vera, where the entire premise of treatment is creating a state of iron deficiency. Are we, actually potentially harming our patients by creating this, state of iron deficiency? And how do we deal with the symptoms like restless leg syndrome, etc., which develop because of the resulting iron deficiency?
I'm very sorry, the volume, could you speak up a little bit? So I couldn't understand your question.
Am I audible, ma'am? I hope.
You are audible. Just speak slowly.
Hello.
Abha, you are audible. Speak slowly.
Hello.
Hello, yes.
I'm sorry, even I'm not able to hear correctly.
All right, we'll come back to you. Dr. Bharat, your question.
Madam, excellent lecture and many new ideas. Thank you for the same. Now in developing countries, particularly in India, anemia is widespread, prevalent, and iron deficiency is the main. Not only adult population, but adolescents, children, and it influences the work performance in children, learning abilities and cognitive performance. So government of India has implemented food fortification programs for distribution of fortified rice with micronutrients like rice, B12, like iron, B12, etc. So, in some areas, particularly tribal population where hemolytic anemias are prevalent, there is some problems, but otherwise it has worked well. So, your thoughts or your suggestions on the same?
Yes, thank you. I think nutritional supplementation of iron is a great idea, and I think it's probably very a good way to prevent the development of iron deficiency. But when there is inborn iron deficiency that results from the mother not giving the newborn enough iron, so we have these. And we see this particularly the peripartum cardiomyopathy study I showed earlier. These are pregnant women from India who, end up with hemoglobins of 4, 5, very low iron levels, and they have no iron stores to give their fetus, and their newborns are born with very little iron to start with. And when you have profound iron deficiency, you're born with profound iron deficiency, dietary iron supplementation isn't going to help, isn't going to be sufficient. And that's the problem. I think dietary iron supplementation is a fantastic way to prevent the development of iron deficiency, but it isn't sufficient to address the iron deficiency that a lot of children are born with because of maternal iron deficiency.
Yeah. Dr. Patodia?
Good evening, madam. Thank you very much for the lecture. I am a medical oncologist, not a hematologist. I practice in a setup where I see nearly 90% GI cancers, and almost all have iron deficiency anemia simply because they are oozing somewhere, either gastric lesions or colonic lesions. And the question which I face on daily basis is, I know that they are iron deficient. But many of our chemotherapies, especially adriamycin-based chemotherapy, we suggest that don't give iron when you are giving adriamycin. Now, this particular setup, thankfully in GI cancers, the adriamycin-based drugs are getting out, but still, in this setup, I know that they are GI deficient. If I'm going to give an adriamycin-based drug, should I replace the iron or not replace the iron?
Thank you. No, thank you. That's a really interesting question. The thing about giving iron to cancer patients is thinking about the patient needs the iron, but so does the tumor. And that is the complex thing to worry about. So we know, for instance, from our collaboration with a group in Michigan, that colorectal cancer cells, for instance, really need their iron, and they go to great lengths to make sure they retain the iron. In fact, colorectal cancers will express their own hepcidin to make sure that they retain all the iron that they can. And that hepcidin production locally is required for the tumor growth and metastasis. So you have the dilemma, and I don't envy you because it's a tricky one. Do you give your patient iron and make them systemically well, but in the process you may also be helping tumor growth? Now, it's probably not very straightforward in patients because there will be a component of iron deficiency caused by blood loss. Yeah. And there's a component of iron deficiency caused by inflammation. And I think the key is to try to establish which of these two groups your patients fall in. Is that a case of absolute iron deficiency caused by blood loss? You would be able to tell those people normally by low ferritin and low TSAT. And if you're able to test for hepcidin, you will also find that they'll have low hepcidin. The other group will be those that have iron deficiency because of a blockade of iron absorption in the gut, because they have high hepcidin and inflammation assets. You'll be able to tell those because they'll have low TSAT, high ferritin, and high hepcidin. And those are the two groups. But then in either way, you would have the dilemma of, well, can I be sure that the iron that I'm giving the patient isn't also going to benefit the tumor?
Unfortunately, hepcidin is not available, at least in my knowledge.
I think TSAT and ferritin would be a good way to tell the absolute from the functional.
Okay. Thank you so much, Dr. Samira. Unfortunately, we have run out of time. There are still many raised hands and there are many questions lying in the chat box, but we will have to stop here. I'm extremely grateful to you for having accepted our invitation, and what a different angle you have given us about this subject. Thank you.
Thank you very much for inviting me. And for those of you who, I'm sorry I couldn't get through to your questions, please feel free to send them to me by email.
They'll do that, sure.
Thank you very much indeed. All the best.
So we'll move on to the second lecture of the day, and that is by Dr. Daniel Hodson. It's my pleasure to introduce him. He is Group Senior Research Fellow, Honorary Consultant Hematologist. Welcome, MRC Cambridge Stem Cell Institute, University of Cambridge, Department of Hematology, the Jeffrey G. Biomedical Center at Cambridge, UK. And he is going to speak to us on molecular profiling in diffuse large B-cell lymphoma challenges and opportunities. So over to you, sir. You have to unmute yourself.
Oh yeah, apologies. Can you hear me now?
Yeah, yes.
Yeah, thank you for that nice introduction and for inviting me. And I'm hoping that in a second you'll be able to see my screen. Does that look?
Yes, perfect.
Perfect. So thanks for inviting me. And what I want to talk about today is molecular profiling in diffuse large B-cell lymphoma. And in particular, I want to talk about really the new genetic subtypes of diffuse large B-cell lymphoma. And I've got a number of titles for this talk. One is sort of, do we need next generation sequencing in lymphoma practice? Another is the one I think I gave you, which is challenges and opportunities. But really it's genetic subtyping of DLBCL. Do we care? And why should we care? So this is the disease, of course, that we're talking about. This is diffuse large B-cell lymphoma, and I'm sure everyone is very familiar with this, but just in one slide to run through some background. This is the commonest form of non-Hodgkin lymphoma. It presents with an incidence of about 70 per million per year, and it presents with rapidly enlarging tumors that are either nodal, like in this case here, mediastinal, retroperitoneal nodes, but often in about a third of cases extranodal. And at the moment, the diagnosis of DLBCL requires a biopsy where we look for histology. So these are this is a diffuse infiltration of large cells, large lymphoid cells, and immunohistochemistry. And in particular, of course, this is CD20 here, confirming that these are B cells. Now, we also, the WHO criteria at the moment also requires a cell of origin assignment and MYC, BCL2, BCL6 status by FISH. And I'll talk a little bit about these, but more I want to talk about where we're heading in the next 5 to 10 years with classification of DLBCL. Staging is by CT or by PET-CT, and first-line treatment is with R-CHOP. And you could argue all day about the different types of chemotherapy or intensification of chemotherapy that you might use for DLBCL, but really the only proven treatment in randomized trials for DLBCL is R-CHOP. And so if I go to the next slide, this is a rather old slide. This is the randomized trial that showed that the addition of rituximab to CHOP improves the overall survival. So on the y-axis, very important, this is a sort of old-fashioned randomized controlled trial. Overall survival on the y-axis, which we rarely see now. Time on the y-axis, and you can see whilst this is significant, you don't need the significance. You can see that there's a clear advantage to the addition of rituximab to CHOP. Now, the reason I show you this very outdated study, which was published more than 20 years ago, 2 decades ago, really is to reinforce that this was the last time that we saw a difference in overall survival with diffuse large B-cell lymphoma for first-line treatment. And so what I've done on the right of the slide is to summarize the major studies that have happened since. So since the study on the left, we've actually now had 17 phase 3 or at least large randomized studies in diffuse large B-cell lymphoma, which can broadly be summarized here. And perhaps something to, if you're going to look at one column, is this column here that says number of patients enrolled. And if you add these numbers together, you get more than 12,000 patients have been enrolled in DLBCL randomized trials since R-CHOP was introduced. But none of these trials has yet led to a change in standard of care first-line therapy. And these trials tried really largely 2 approaches. One is intensification of chemotherapy, but the other is the incorporation of what I call targeted or molecularly targeted drugs. And all of these molecularly targeted drugs looked really great when they were tried in cell lines or animal models and when they were published in their Cancer Cell or Nature papers. And the question that we really have to ask ourselves is, why are we not making more progress? So why has the last 2 decades of DLBCL been really so disappointing in terms of clinical trials? And this is a rather out-of-date slide, but it serves to make 2 points. So firstly is that we know a lot about the biology of DLBCL, and we've learned a lot in the last 20 years. So we know about the oncogenic pathways that these tumors require in order to survive. And then the second point I want to make is that there is no shortage of targeted drugs. So I've just labeled a scattering of drugs here, but I mean, there's probably 2, 3, 4 times this many. And if you start to think about how we might use these in combinations, then you get into very large numbers of drugs that we might want to trial in DLBCL. So it's not that we're short of drugs, and it's not that we don't understand the biology, but this, I think, is why we are not making more progress in DLBCL trials, which is that basically we have these magic bullets, but we are just shooting them as if we were blindfolded. And perhaps one sort of take-home message from this study, or from this presentation that I might want to leave you with, is the notion that if we're going to use biologically targeted therapies, that these need to be targeted at the biology. And perhaps a second message I want to leave you with is that there are, in my opinion, no biologically agnostic therapies. There are only biologically agnostic trials. And the reason I say this is because we have had some success in DLBCL in recent years in using what people tend to call biology agnostic therapies. Examples being CAR T, where it hits CD19 and it doesn't require any specific biology. Or polatuzumab, which hits CD79B, doesn't require any specific biology. But these are not really biology agnostic. And when you look at these, when you look at CAR T therapy, it's very clear that the activity of CAR T cells depends on the p53 status of the tumor. And it may well be, if we look further, and we just haven't done this yet, that it depends on a whole lot more biology. Polatuzumab, very similar. It's apparently labeled as a biology agnostic therapy, but when you look in the supplementary information and you see how did patients respond in terms of subgroups, in terms of GCB, ABC, it's very clear that polatuzumab activity is restricted to the ABCs. So these are not biology agnostic therapies. So why is this such a big deal for DLBCL? And the reason that I think this is a big deal is that DLBCL at the sort of molecular biology level comprises multiple different molecular diseases. So that's not a new statement, and we've known that DLBCL is more than one disease for about for 22 years, in fact. So this is the cell of origin story, and I'm just going to summarize this in one slide and point out maybe a few things that might be relevant as we go forward to alternative classifications. So this was driven by developments in technology around about the turn of the millennium, when it became possible to do global whole transcriptome profiling. In those days, that was done by microarray. If we were doing this now, we'd do it by RNA sequencing. But this showed two dominant clusters based on messenger RNA abundance in microarray, and based on their similarities to germinal center B cells or B cells that were activated in vitro, these became known as the ABC subtype and the GCB subtype. And this was described initially in 2000 by Ashilizaday and Lou Stout's group. And then there was a gap. There was 17 years before this was incorporated into the WHO classification. And then unfortunately, within a year of it really being accepted, an alternative classification was proposed in 2018. But there's a few things that I just want to reinforce. So I think I would argue that the cell of origin classification probably had little practical benefit in the clinic. So I don't think we at the moment treat ABCs, GCBs differently, and I think we only make a cursory effort to distinguish the two. But what I think it was invaluable for is as a framework to dissect the biology of DLBCL, and pretty much everything we've learned in terms of DLBCL biology when it comes to the epigenetic corruption and chromatin corruption that we see in the GCBs, and the activation of NF-kappa B, toll-like receptor signaling, and B-cell receptor signaling in ABC DLBCL, all of that has arisen by being able to compare the biology of these two subtypes. So I think it really, the legacy of this transcriptional classification is the biology that we've learned from it, not so much the prognosis that it describes. But it's also worth thinking, why did it take 17 years to go from description through to incorporation into the WHO classification? And I think that was for a number of reasons, and I think you could transfer these to any modern classification that we're trying to apply. So one is that it was simply difficult to perform transcriptional profiling in routine clinical labs 20 years ago, and I think it probably is quite difficult to perform it in most labs now. And as a result, this led to a series of oversimplistic proxy assays that I think over 20 years we've learned probably have relatively limited accuracy and therefore relatively limited value. And I'm talking about the sort of immunohistochemical approaches to try and reproduce the cell of origin classification. I think also there was confusion about the prognostic benefit versus the biological implications. And I think this is a critical thing going forward, that a classification doesn't have to give you prognostic information in order to provide important biological information. So there are just some sort of thoughts about historical classifications that we might want to carry forward when we're thinking about newer classifications. And by newer classifications, I'm really referring to these 4 studies. So in 2018, there were 2 studies, one in Nature Medicine from Margaret Shipp's group at Harvard with 304 patients profiled. Shortly afterwards, there was one in the New England Journal of Medicine from Lou Stout's group at the NCI with 574 cases profiled. And then a couple of years later, another study from the NCI really was a reanalysis of all of the published data so far. And then about the same time, our group in the United Kingdom published a targeted sequencing study of just under 1,000 DLBCLs, again, asking the same question in all of these studies, which is, are there molecular subtypes or genetic subtypes of DLBCL? And I won't go into these in this particular slide in too much detail, but just to say that this is how the data inevitably is presented. And if you're shown these, just to sort of summarize what this means, but going up on the y-axis here, you can see a list of genes, and the colors usually mean that that gene is mutated. And if it's in blue versus red, it refers to different types of mutations, which are usually sort of amplification versus single nucleotide variants versus nonsense mutations, etc. And each vertical line is a patient. So this is 570 patients lined up across here. This is 300 patients lined up across here. And you can see that they apparently cluster into subtypes of patients that look similar in terms of the mutation profile that they have. So before we go into this, I want to point out that I look at this with 2 hats. So I look at this partly as a clinician, but also, of course, as a biologist, and I run a research group in the University of Cambridge, and I split my time between treating patients in the hospital, but also researching the biology of DLBCL. And so I'm well aware that there are two sort of different camps of thought here. And on one side, you have the sequencing guys who say, you know, the problem is now solved. This is the final answer to heterogeneity. This will now tell us how to treat all our patients. But on the other side, we have a bunch of perhaps sort of wiser, more senior clinicians who say, I've seen this all before with cell of origin profiling. I retire in 18 years, therefore I can probably safely ignore this. This is not practical. You'll never bring this into clinical work. So we have to, I think, answer some questions. And these are the questions that I want to address in the next sort of 25 minutes. So I've said that I want to provide balanced answers, and I want to provide balanced answers that come from both a biological sort of science perspective, but also from a practicing clinician perspective. So these are the 4 questions. So firstly, what are the subtypes and why do we believe that they're real? Secondly, do these genetic classification systems agree at the individual patient level? Thirdly, do they tell us anything useful about prognosis? And fourthly, do they tell us how to treat patients? So to answer these questions, we set out in the UK to perform the largest study to date of genetic subtyping. And this was a big collaboration across the United Kingdom. And I've just pointed out, here's a map of the UK. Here's Cambridge in this dot here over on the eastern side of the country. But really all our patients came from a county called Yorkshire, which is one of the largest counties in the United Kingdom. And the unique thing about Yorkshire is every single patient diagnosed with blood cancer in Yorkshire is diagnosed at a single central pathology institution in Leeds. And every patient is also enrolled into a heme malignancies registry study. So this means that we're not subjected to some of the same bias that clinical trials are, where they tend to recruit the good risk patients, or pathology referral centres, where they tend to recruit the sort of the funny histology and the poor risk patients. But it means we recruited consecutive patients, I think between 2002 and 2012, every patient diagnosed with DLBCL in Yorkshire went into this study, so it's consecutive patients. We extracted DNA from FFPE biopsies, which is different to the two US studies and perhaps makes us a little bit more relevant to clinical practice. And we performed a targeted sequencing panel. So we assessed mutations in 293 genes. And we sequenced just under 1,000 cases of first-line DLBCL. We then used a completely different statistical modeling process to assess whether there are genetic subgroups. And the important thing to point out is that really at every stage in our study, we are therefore different to the two previous US studies. And so the question is, did we reach the same conclusion or did we reach different conclusions? And this is broadly a summary of what we found. So I've lined it up a little bit like the two studies I've shown before. So going down here is 60 odd genes that are most frequently mutated. This is the cell of origin classification on the bottom. These are, and each vertical line represents a patient. And our clustering formed patients into 6 genetic clusters, which we named at the top here based on the most frequently mutated genes, with an unclassified group over here on the left. But all of our clusters, the exciting thing, could be mapped to one of the Harvard or NCI genetic clusters. And I think we didn't set out to reproduce the findings of the other two groups. In some ways, we would like to have made sort of novel findings of our own. But what was really striking is the remarkable level of consensus that we now have between our study in the UK, the Harvard study and the NCI study. And really, we very much converged on almost identical conclusions about the nature of genetic subtypes of DLBCL. And this is a level of consensus across 3 big studies that's really quite rare to see in academic medicine. And, you know, if we look back to the days of transcriptional profiling and cell of origin classifications, there simply wasn't the degree of consensus then that we see now around these genetic subtypes. The second thing that I think is very interesting is that once we started to identify these subtypes, both our study and the other studies made the observation that these really look like, each of these subtypes look like types of lymphoma that we already know about. So this is all DLBCL, but we have DLBCL that carries the same mutational profiles as follicular lymphoma. We also have DLBCL that carries the same mutations of or the same mutational profile as extranodal lymphomas, so primary CNS, testis, breast lymphoma. We have a genetic subtype of DLBCL that looks just like primary mediastinal B-cell lymphoma. We have a genetic subtype of DLBCL that looks like marginal zone lymphoma, and another one that looks like what we know about double hit lymphoma. So all of this, I think, starts to blur the boundaries between our existing clinical classifications and what might in the future be biologically based classifications. So this is my conceptual map of diffuse large B-cell lymphoma classification. And what I've done is I've labeled in white or black in the middle the names of genes that are most commonly mutated within these subtypes. And the color density reflects the confidence of the classification. And so you can see here that there's 6 or 7 different subtypes. And just to run through each of these briefly, if we start in the top left here with in blue, you can see what we call the MCD subtype. So that was named MCD in the NCI study for MIDI88 CD79B. The Harvard study called this C5, and we called this MYD88 because that was the most enriched mutation. But this was a genetic subtype that leapt out of every single of the 3 studies. And all 3 studies agree almost precisely about the nature of this study. When we look by gene expression, this is like a sort of pure ABC DLBCL. And this is heavily enriched for cases of CNS lymphoma, testis, breast lymphoma, and shares very similar mutations. So this is a very discrete type of diffuse large B-cell lymphoma that really is completely different to all the others. At the other end of the spectrum, we have what's known as EZB lymphoma or C3 lymphoma, or we called it BCL2, again, because that was the most, BCL2 translocation was the most enriched genetic alteration in this subtype. And again, this was distinct in all 3 of the studies, and we agree almost exactly on the nature of the mutations in this subtype. But this is basically the high-grade cousin of follicular lymphoma. It's exactly the same mutational profile that follicular lymphoma has. And in fact, 12% of the cases in our study were transformed follicular lymphoma. Almost all the transformed follicular lymphomas clustered into this group. And more than a quarter of cases when we did a bone marrow, we were able to find previously unknown or unrecognized follicular lymphoma in the bone marrow of these cases. So this is a kind of high-grade version of follicular lymphoma. There's a group down here at the bottom left in green called BN2, or Harvard called it C1, we called it Notch2. And this again was distinct in every analysis. And it's equally split between GCB, ABC, and unclassified, and really totally unknown before we started the genetic profiling. But this looks exactly like marginal zone lymphoma when we look at the mutations. We didn't find cases that had transformed from marginal zone into this subtype of DLBCL, but it's clear that the biology is shared. There's a 4th group that again came out of every study, which is enriched in JAK, STAT, and TET2 mutations. We found that this was similar to primary mediastinal B-cell lymphoma in its biology. And in our study, perhaps because of the bigger number of cases, we actually managed to divide this into 2 different subtypes. Then there's a Notch1 subtype, which looks a little bit like Richter's syndrome, has a poor prognosis, and a TP53 mutant aneuploidy group, which has no specific mutations beyond P53, but is enriched for really widespread aneuploidy across the genome. So that's my sort of conceptual map. So if we move to looking at some of these questions, so if we can address question 1, which is what are the subtypes and do we believe they're real? I've been through the subtypes, but I just want to really reinforce the remarkable level of consensus between independent studies. And also that gene expression profiling of these studies supports that they really rely on different oncogenic pathways. So we agree on the groups as a whole, but do we agree which patients go into which classification? And so these rather complicated plots then allow us to line up 2 different studies. So there's our classification at the top from the UK study, and then we reclassified it using one of the US studies at the bottom. And it looks at first glance like a lot of stuff is moving around. But actually, what's happening is that cases are moving in and out of the unclassified group. So really what I conclude from this is that the greatest source of variability comes from whether a case is classified at all, rather than movement between different classes. And then the only exception to this is when we compare the 2 US studies, is what happens to this C2 or what they call A53, the aneuploidy group that I talked about, P53 aneuploidy. And I think this is the group where there is not consensus across studies. But everything else, I think there is a real high level of consensus at the individual patient basis. So going back to this conceptual map, what I just want to reinforce is that a patient could land anywhere on this map. And a patient here will be classified with very high confidence by every classifier as being in this subtype. Whereas a patient here might be classified as unclassified by one group, but might sneak into this classification in another group. And that's what I mean by placing the boundaries in different places. So to answer question 2, do the classification systems agree at the individual patient level? And I think there is very strong agreement for most clusters. The only caveat being the A53 group. And I think the main variability is whether a patient is classified at all, or whether, rather than being misclassified between cases. So question 3, do these genetic subtypes tell us about prognosis? So this is the NCI, the US publication, and it looks like there's a few things that you can immediately notice. So one is that there's a red group here called ST2, that's the JAK-STAT mutant enrichment group, that do really very well. And when you look across the 3 different studies, this is the 5-year overall survival, and I've just summarized the survival of the analogous subgroup. So if you look at the red in the NCI study, the red in the Harvard study, and the red and orange, which are the 2 in our study, you can see that these always actually are doing pretty well. At the other end of the scale, you can see some poor risk groups. So you can see that the green always does badly. So this is notch 1 here. This is notch 1 in our study. Again, it's always one of the worst overall survivals. But then the blue, MCD, looks like it does particularly badly here. But MCD doesn't do particularly badly in the Harvard study, and MCD doesn't do particularly badly in our study. And so I think this is because the patient mix is different across studies, and I think this is a really important point. So we looked at this MCD group in our study, and we looked at it in 2 different contexts. So we looked at it in patients that we had treated with what are called R-CHOP-like, which is this sort of slightly vague terminology that retrospective studies tend to use, meaning anyone that had sort of rituximab plus some kind of chemotherapy. So R-CHOP-like, you can see that that MCD group, we called it MIDI88, really does quite badly. But when we restrict to full dose R-CHOP, so these are patients that had proper R-CHOP with no modifications, suddenly that group disappears into the mix of everything else. And I think the thing to point out here is that these genetic profiles are not totally independent from patient age, frailty, and other clinical risk factors. And we can see this from history as well. So if we go back here to the early days of double-hit lymphoma. So here's a publication on double-hit lymphoma from 2009 when it was sort of first described. And you can see the survival, this is overall survival of these patients, is absolutely terrible. Whereas a few years later, or 6 years later, in the context of clinical trials when they looked at double-hit patients, the prognosis of double-hit lymphoma seems to be really not quite so dismal at all. So why is this? And I simply think that this is because in retrospective studies and in clinical studies, we have 3 different groups of patients. So we have patients with absolutely terrible disease like this one. So these patients get referred to pathology referral centers. They accumulate these biopsies in pathology archives. So when we interrogate pathology archives, for instance, when we say, right, what happened to all the double-hit lymphoma patients that we diagnosed in 2009, or what happened to all the MCD patients enrolled in our study, in our retrospective pathology study, you get a really inflated bad outcome. In contrast, we have lots of fit patients, and these go in clinical trials. But then we also have these super fit patients who then travel miles and miles to go in clinical trials and receive more aggressive therapy. And I think this patient fitness is a real issue when we look at the outcome of genetic subtypes or other molecular subtypes when these are accounted either from clinical trials or from retrospective pathology archives. So that was one thing that made our study different. We enrolled, as I said, consecutive patients regardless of their treatment, and then we were able to restrict them to those that had R-CHOP. So this, I think, is the sort of final answer on prognosis. And what we've got here is overall survival, and we've got the same patients classified by 2 genetic classifiers. This is the LymphGen classifier on the left, that's the NCI classification. And we've got the modified HMRN classification, that's the UK one on the right. And you can see that the survivals look very, very similar between the left and the right plots. But the two key things that are bad are Notch1 cases do badly, and these MYC-driven cases, and we call them different things in different studies, but it's the same patients. So it's those that are the sort of easy B subtype with MYC translocation or with MYC transcriptional profiling, and they do badly. But the rest are somewhere in the middle. So the answer to question 3, do they tell us about prognosis, is that the ST2 subtype is good, MYC and Notch1 are bad, MCD is probably bad, but I think we have to be very aware of recruitment bias into these retrospective studies. And I'm going to really reserve judgment until we've accrued more patients in the future. I would caution people to be aware of studies that describe R-CHOP-like patients. But the most important thing perhaps is to say that prognosis is not the reason why I think we should be doing molecular subtyping. And I think molecular subtyping is all about the biology. So, do these subtypes tell us how to treat patients? And you see sort of studies like this in lots of reviews where they say, now that we understand the biology, we know what treatment the patient should have. But I disagree with that, and I think that we just simply can't predict from the genetics at the moment or from our current preclinical models. But I do think that molecular profiling will allow us to interpret future studies much better than we can at the moment. And I'll give you 2 examples of that. So this data comes from the Remodel B study. So this was a phase 3 study of an R-CHOP plus or minus Velcade in first-line DLBCL. It was about 800 patients, 900 patients, something like that. And this was a failed study. Across the board, when you look at DLBCL as a whole, we saw no differences in survival. But if you take this transcriptional subtype, which actually I haven't talked about yet, but is called molecular high-grade lymphoma in the UK study, in the Vancouver study, they call it double-hit SIG. And basically, this is similar to double-hit lymphoma identified by FISH. It's all within the germinal center subtype. There's this molecular or MYC-driven group, molecular high-grade. And there's some, but imperfect overlap with FISH. It has a bad prognosis, but what's interesting is when we restrict the analysis of this study just to those patients with molecular high-grade lymphoma and ask, did the addition of Velcade make any difference to them? You can see that there starts to emerge this trend towards better survival in the molecular high-grade patients that received Velcade. So this is a hypothesis, or this is hypothesis generating, and this will now be taken forward and tested in a prospective trial. But the point I want to make is when we start to look for molecular subtypes in trials that have otherwise failed, we start to see molecular subtype specific responses to these targeted drugs. Here's another example. This was the Phoenix trial. So you probably all know the Phoenix trial was published quite recently, and it was a randomized phase 3 trial of R-CHOP plus either ibrutinib or placebo. And across the board on the left, you can see here, there was no difference in survival between the placebo and the ibrutinib arms. But more recently, the Stout group has been back to this study and they've reanalyzed the patients. And what they've done is they've applied the LymphGen genetic classifier that I've shown you earlier on, and they've said across individual subtypes, how did patients respond? And it's quite remarkable. So here is the MCD subtype in blue. In dark blue are the patients who received ibrutinib. In light blue, the patients who received R-CHOP plus placebo. And you can see that this is highly significant. We see a similar result again for the Notch1 group, where the ibrutinib arm at least appears and achieves statistical significance to be doing much, much better than the placebo arm. And then here's in pink is younger patients that don't have Notch1 or MCD subtype. And you can see there's a little bit of difference, but not much. So my conclusions from this are that different genetic subgroups respond differently to biologically targeted groups, but also that results are not always predictable from preclinical data. So we didn't expect that the molecular high-grade group would respond to Velcade. We didn't expect Notch1 genetic subgroup would respond to ibrutinib. And this is one of the reasons why I think we're not ready yet to use genetic subtypes to determine what treatment patients should get. But I think we are ready to use genetic subtypes and molecular profiling in general to interrogate our studies so that we don't any longer treat DLBCL as a single disease in studies. So do they tell us how to treat? Not yet. But they will allow us to design and interpret better clinical trials. And the point I want to make is that when we look for subtype-specific responses, we find them. So I'm going to talk now in the final couple of slides about what do we need to do for next-generation sequencing in clinical trials, and then what kind of next-generation sequencing should we do in routine practice. So this is what I think we should be doing in clinical trials. So in the middle here in this pie, I put what I think are the 3 reasons to be doing molecular profiling in clinical trials. So first of all, if we take this slice here, I've put to apply current and future molecular classifications. And I think this is really important. I don't think we have the final answer on the molecular classification of DLBCL. And I think a trial enrolling now that maybe takes 5 years of follow-up and a few more years to publish will probably be published at a time when the molecular classifications have evolved and refined. And so I put here that we should expect continued evolution in genetic classification of DLBCL. So I think there's a whole bunch of undiscovered subtypes that probably sit in the unclassified group, and I think we will refine and reach even more consensus between the existing classification systems. But exactly what genes and what mutations that's going to depend upon, I think is not known yet. And so I think future-proofing is essential here. So I think any clinical trial for DLBCL needs to be future-proofed and able to apply future molecular classifications. But these will be built on mutations and gene expression profiling. So secondly, I think trials have to be able to interface with discovery science. So in my lab, we run CRISPR screens, we discover unexpected pathways and genes and mutations that confer sensitivity or resistance to particular drugs. But at the moment, it's almost impossible to go back to the drug trials and say, so did the patients with that mutation respond? Largely because they don't have that data available. So I think that the molecular determinants of response and resistance and interfacing with discovery science is critical. And I think any trial that is unable to do that because they can't provide the molecular data should really be seen as a wasted trial. So I think we need to be able to test genetic hypotheses that emerge from future discovery science. So like this, I think trials have to be future-proofed against hypotheses that arise from experiments that we haven't yet done in the lab. And then finally, I think trials need to contribute to building a molecular knowledge base. So we're very good at banking tissue at the moment, at least in the UK. We do big studies, we bank tissue on everyone, we put it in the freezer, and then we say, great, we've banked tissue from 1,000 patients. But I think we need to reflexly now be profiling the gene expression and mutations of these patients and building up what I hope across the world could be a joined up molecular database, so that people can start to ask these type of questions of studies as they are published in real time. So I think for the purposes of clinical trials, I think all patients should have a full exome and a full transcriptome, and I think this should be paired with their clinical data, and this should be made available at the time that the trial is published. And whilst all studies do some kind of molecular profiling, I think we're yet to see this kind of really user-friendly publication of the data, but I hope that will change in the future. So as we get to the end, what about routine diagnostic labs? So I think there are real challenges to performing next-generation sequencing and genetic subtyping in diagnostic labs. And I think these are there are 3 real challenges. So first is sample-related challenges. So I don't quite know in India, but in the UK, we've moved very much against the will of hematologists and pathologists towards tiny core biopsies, often with a tiny bit of lymphoma in there. And usually by the time the pathologist has finished with this, there's simply nothing left for molecular analysis. And we have to find ways to overcome that. And that might be setting biopsies in different blocks and reserving one for molecular analysis, Or it might, as future technology involve using circulating tumor DNA, which we can extract from plasma, from a blood test, as the source of tumor DNA for molecular profiling. And I increasingly think that circulating tumor DNA is going to be how we do our molecular profiling in real patients. The other challenges are just simply logistic. So getting an FFPE block from a pathologist in one hospital to a sequencing laboratory in another hospital is surprisingly challenging. So we've been running a study in Cambridge in the UK where we profile the genetics of all our new patients. And this is just perhaps the most disappointing part of this study. And this is the time from the patient's diagnosis to the time that we get the block in our DNA sequencing lab, in days. And the average time, despite trying very, very hard to get this and do this in real time, in a clinically useful turnaround, is 74 days. So however quickly we can do the sequencing and the analysis, you've got to add 74 days of logistics at the moment until we iron out these logistical challenges. And then finally, there's technical challenges. So what sequencing platform do we use? Do we do targeted sequencing, exome sequencing? How do we call variants? And the way we call variants and driver mutations is hugely different at the moment across the big academic studies. So here, for instance, is SOX1 mutations, and I've just put the 4 big studies. One study doesn't call any, same with DTX1 mutations. A different study, again, doesn't call any. It's not that they didn't find them, it's just that the hugely complicated filtering process they put into their bioinformatic pipelines basically say these aren't important and they get rid of them. So there has to be some kind of standardization across clinical labs and some way that we can organize the quality management of this process. So these problems won't be solved in academic studies. And I think these will only be solved when routine labs basically start genetic profiling and start working through these problems. And the reason I think we have to start working through these problems and building the infrastructure now is that I think it's only a matter of time before the next wave of DLBCL first-line studies, where there will be extensive molecular profiling, start to identify subtype-specific therapies. And we risk, if we don't start some form of genetic profiling now, we risk being in the position where in 5 years, we have a whole list of subtype-specific responses to targeted therapy, but we have no way in the real world to subtype these patients. So the key thing is, I think we have to break this vicious cycle. So at the moment, I think we say we don't quite understand what the implications of next generation sequencing mean for clinical management, and also it's difficult, and therefore we don't do the NGS. And then because we don't do the NGS, we never solve the problem of how to actually do it or what it means. But I think if we were to break through this and make NGS more routine in the diagnosis of lymphoma, we will overcome these feasibility and technical issues, and we will start to build a molecular knowledge base or a molecular knowledge bank of both trial and real-world data. And that will then help us understand exactly what this means and allow us to interface with discovery science. So essentially, I have 4 or 5 final conclusions in summary of this talk. So first of all, the key point is that DLBCL comprises distinct molecular diseases. It's not one disease. And I think recognizing this heterogeneity will be essential to therapeutic progress in DLBCL. Second, I think there's now remarkable consensus on the genetic subtypes across 3 or 4 major studies. And I think that suggests that whilst this might not be the final answer, this will at least be the basis for resolving the heterogeneity. The next important point is I think these classifications will continue to evolve, but the final answer is likely to combine genetic and gene expression information. And therefore, I think trials now know what they need to do to future-proof themselves against these changes. I think comprehensive genetic and transcriptomic profiling really must be mandatory for drug trials, and I think trials that don't include that could really be considered a wasted opportunity. And finally, I think the role in routine practice is much less clear, but for the reasons I've articulated, I think now is the time we have to start building the infrastructure for real-time molecular profiling. So I will stop there, and what I'm hoping is that there can be some questions and perhaps some discussion. So thank you very much.
Thank you, Dr. Hodson. That was fantastic. What a piece of work and what clarity of thoughts. So we have some colleagues who want to ask questions. Dr. Shilpa.
Hi, Daniel. Thanks for the wonderful talk. My question is that you have had studies with the genetic tests which are done, the molecular profiling prior to chemotherapy. But I just wanted to know whether, have you seen such responses in people who have undergone CAR T, like prior to CAR T, you've had some molecular profiling and you have seen that such molecular profiling does not work for these patients post-CAR T? I mean, they respond poorly to CAR T? Because CAR T is an expensive thing.
Absolutely, yeah. So I think, and I think but studies often say, oh, we can't afford to do the exome sequencing or the genetic profiling. But, the cost of CAR T is so expensive that I think that really justifies really very extensive molecular profiling to work out who will respond and who won't. And that hasn't been well done. So there've been a lot of small studies where local, like one institution has sequenced their 30 patients they did that year, something like that. And they tend not to show any correlation between genetic subtypes and CAR T response. But having said that, I think it's very difficult to answer that question with such a small number of patients. And there's so much else going on in terms of the diversity of patient clinical risk factors, and that I think it just makes it impossible to really dissect out the contribution of the genomic subtypes. The only thing that I think is, or perhaps the best data, is that there was a JCO paper, I think maybe this year, possibly last year, where they looked at a selection of mutations. I mean, it wasn't a lot of mutations, but one of them they looked at was a TP53 mutation. And it was quite clear that patients that are TP53 mutant do less well in response to CAR T. But had they done sort of full exome, and I wouldn't be surprised at all if there's a whole load of other molecular determinants. So I think in answer to your question, people are starting to look, but we really don't know the full answer to that question yet.
Thank you.
Thank you. Dr. Rajat.
Yes, Dr. Hodson, fantastic talk. I'm a pediatric oncologist. So my question to you, whatever you talked about, does that apply to pediatrics? The majority of our children have buckets. They actually do very well with FABLMB protocol. And the second question is, we have just started doing NGS profiling for our lymphomas as well in pediatrics. Do you use a commercial panel, or you devise your own panel for NGS screening?
Yeah, so good question. So the first question, I don't think people have really applied these NGS or genetic subtypes to pediatric lymphomas, but I suspect, what we can see is that there's a shift in the genetic subtypes as you go from young adults to old adults, and I suspect children may be at the sort of extension of the young adults. So for instance, the MCD type, which is the sort of MYD88 CD79B mutant type, becomes more and more common as patients get older. So it's rare in young adults, it's really much more common in older adults. And it may just be that we'll see more of the sort of ST2 type and the EZB type amongst the DLBCLs of children, and of course Burkitt as well. So Burkitt is separate to these DLBCL subtypes, but again, I think we don't know. And my sort of argument for doing more routine profiling and finding some way to pool this data across countries and across institutions in a way that it can be pulled together is that we'll start to answer these questions that maybe an individual institution or an individual study can't answer when you're talking about relatively rare tumors like DLBCL in children. And then in terms of your second question, so which panel do we use? So we have our own panel, which is a sort of DLBCL specific panel, which is different to any of the commercial ones. I mean, broadly, the driver genes, I think, are fairly defined. There's about 100 to 150 genes that are defined, and they're what, you know, with those you can confidently assign to one of the genetic subtypes. I'm not actually sure if you can buy this from a commercial provider, but it's relatively easy these days to make your own focus panel, and you go to Twist Bioscience or something like that and say, right, here's the 150 genes we want on our panel, and they just make it for you.
Okay, thank you.
Thank you. Take one question from the audience. Dr. Raj Warrior wants to know, does presence of cancer predisposition syndromes like ataxia telangiectasia or immunodeficiency syndromes alter the prognosis or affect the molecular markers?
So again, I think for all of these questions, I'm giving a slightly long-winded way of saying I don't really know. But I think, again, we haven't sequenced enough of these people. But my intuitive thought is that both of those things, you know, sort of DNA damage repair and, you know, primary immunodeficiencies will make a big difference in terms of altering the outcome. We know, for instance, that when we sequence cases that have an immune defect, that they have fewer mutations, suggesting that the barrier to transform to a malignant disease is not as high as in someone with a functioning immune system. But quite how these molecular subtypes apply to people with primary immune deficiencies or with DNA damage repair defects, I don't think we've yet looked at.
Thank you. One more question is from Dr. Sham regarding present status of doing ctDNA in DLBCL.
Yeah, so this is something I'm very excited about. And I think ctDNA can broadly answer two important questions. So one is it can be a source of DNA when we struggle to get that from a core biopsy to do molecular profiling. Now, that's not as straightforward as it sounds. And when we compare the molecular profiling we get from a tumor biopsy with the same patient where we use ctDNA, the results overlap about 60% of the mutations we find in both. But the question, of course, is which is the right answer? Because when you biopsy a single site with a core biopsy, you don't take into account the molecular heterogeneity of a tumor across a patient. And it may well be that the tumor in the bone marrow is slightly genetically different in terms of the subclones to the axillary lymph node. And of course, when you biopsy one, you get one answer, whereas when you take ctDNA and profile that, you get a combined answer. So in that sense, it might be more accurate, but you lose the low frequency subclonal variants. I think that's a definite role for ctDNA. The more exciting role for ctDNA, which is considerably more complicated, is to use it to assess response to chemotherapy. So to monitor the tumor burden in the plasma at intervals after chemotherapy. And it's quite clear from published studies, but also from work that we've been doing here, that after one course of R-CHOP, the tumor burden in the plasma drops by at least 1,000-fold. Really quite remarkably. But trying to distinguish whether it drops 100-fold, 1,000-fold, or 10,000-fold is actually quite difficult to do, because you're looking at tumor variant fractions down at 0.001%. And at that level, calling what's a real mutation with what's background noise is very difficult. So I think there's going to be a definite role for ctDNA in both those two, but I think standardizing these assays is going to be key. And I think probably we're going to enter a phase like we have with interim PET scanning, where ctDNA, I mean, interim PET scanning has been around for 10 years, and we keep sort of saying it's predictive, it's not predictive. Probably it determines prognosis, but we're not quite sure what to do about it. And I think we're probably going to have to now go through that with ctDNA. But I suspect it has enormous promise, but I think, will it be influencing anybody's practice next year? Probably not.
Thank you. Last question from Dr. Hari Menon.
Yeah. I was wondering, with all the kind of profiling of the DLBCL, and we need to sort of apply this to clinical trials. Is it time for us to jump the bandwagon and implement based on pathways that are predominant for that, and use those drugs in a relevant fashion to hasten understanding of the outcomes of these drugs for these lymphomas? Because let us look at the example of ibrutinib and also several other small molecules which had subset benefits, and we do understand that most of them are in the ABC subtypes. So is there, I mean, you have made it amply clear in your study, but I just wanted to understand whether that's the way to go ahead in the future.
So do you mean in clinical trials, or do you mean?
Yes.
Yeah, okay. So I think in routine practice, no. And the reason I think no in routine practice, I know you didn't ask this question, but the reason I think no in routine practice is because if you look at what's happened with double hit lymphoma, so double hit lymphoma was described, I don't know, 15 years ago, something like that. Everyone said it's dismal. Everyone who has R-CHOP with double hit lymphoma will die. And then there were some sort of single arm studies where non-randomized studies where patients had intensified chemotherapy and they seemed to do better than historical patients from publications. And then it sort of became fashion that everyone with double hit lymphoma gets intensified therapy. And people became so entrenched on either this is not proven or this is clearly gold standard that it became impossible to do the randomized control trial. And so I think we have to really make sure we don't get to a position where patients with MCD lymphoma get ibrutinib without properly testing that in a trial. Otherwise, we'll find ourselves in 10 years giving it to everyone and then discovering that maybe actually the toxicity is greater than the benefit, and why did we do that for 10 years? So I really don't want to get in that position. When it comes to clinical trials, I think we probably are in a position to make some hypotheses. But the two cases that I showed, and the reason I really stressed it, was that we didn't expect Notch1 cases to respond to ibrutinib, and we didn't expect molecular high grade to respond to Velcade. So if you don't do some kind of sort of screening trial at first, you would miss those unexpected responses that you don't predict from the preclinical data. So the way I would really see a phase 3 trial going in the future is that drugs are maybe rotated through, and a drug comes in, is applied to everyone, and then you start to see a signal and you say, we have some evidence that this subtype responds to this drug. And then at that stage, you then increase the number of patients in those subtypes that get that drug. So a sort of adaptive trial. Whereas if you say, we really don't think, you have to kick these drugs out quite early, I suspect. But if you say, right, in this subtype, we're just not seeing any signal with this drug, then you move in the next drug to that subtype. But I think that kind of adaptive trial that tests more than one drug at the same time in different patients in the same trial is the way we have to go.
Thank you.
Thank you, Dr. Hodson.
And a wonderful presentation on a very complex topic. I understood a lot. Thank you, Dr. Hodson.
Great, thank you very much.
Yeah.
Thank you, Dr. Hodson. On behalf of Mumbai Hematology Group, we are grateful to you for having spared your time. What a lovely talk and what a piece of information. Everybody benefited. Thank you very much.
Wonderful. Thank you so much for inviting me.
Bye-bye.
Okay, bye-bye.
So we go to the last lecture of the day, and that's from Professor Dr. Charlotte Niemeyer from Germany. She has been to Mumbai for a couple of our meetings. She's also spoken to us in the webinars. She's Medical Director, Medical Center, University of Freiburg, Germany, Department of Pediatric Hematology and Oncology. And she's going to speak to us on pediatric versus adult myelodysplastic syndrome. Over to you.
Actually, a very important topic. I just keep it on.
Okay, good afternoon. Can you see my slides?
Yes.
You see them full screen?
Yes.
Perfect. Thank you very much indeed. I very much appreciate your kind invitation because I have very fond memories of my time when I was invited to Mumbai and had the possibility to meet some of you. But this is the wrong slide. Hang on, sorry, I have to go back. This is a mistake. Sorry about this. One second.
Sure.
Hang on. Okay. One more time. Here we go. You have the right title now.
Yeah.
Yeah, I borrowed a few slides last minute from another talk because I said I have 40 minutes. I wanted to be complete. Thank you very much again. The topic today is pediatric versus adult myelodysplastic syndrome. These are my disclosures. So depicted here is the incidence rate of MDS in the different age groups, data from the NIH SEERS database, and I think it's very obvious that MDS is a disease of older individuals. Now in pediatrics, the annual incidence is 1.8 per million under the age of 14 years, or 4 to 5% of all childhood leukemia. In fact, only 6% of MDS is diagnosed in individuals below the age of 50. But as you appreciate, there are also some small children diagnosed of MDS between the age of 1 to 2. Now, thinking about differences between pediatric versus adult myelodysplastic syndrome, you have to consider the nature of the disorder. So in pediatrics, you have more germline predisposition. In fact, you may consider that most of these disorders are actually genetic predispositions. You have perturbed fetal hematopoiesis, perturbed postnatal hematopoiesis, versus in the adult, you're confronted with an aging hematopoiesis. Also, the nature of the host is different. There are young individuals in pediatrics with quite surprising plasticity of hematopoiesis, especially in very young children. And also, these young individuals can tolerate more intensive chemotherapy and hematopoietic stem cell transplantation. Thus, there might be a different classification than in MDS in adults, where often you just discuss palliative therapy. Now, I want to start with 3 examples of perturbed fetal hematopoiesis. And my first example is the natural history and pathogenesis of transient abnormal myelopoiesis and myeloid leukemia of Down syndrome. In the fetal liver of an individual with trisomy 21, there is pronounced proliferation of erythropoietic and megakaryocytic progenitors. So with these biased truncating mutations in GATA1 occur, and the child either develops prenatally or postnatally what we call a transient abnormal myelopoiesis. So it's driven by often large GATA1 clones. In 90% of cases, there is clonal extinction, but about in 10% of cases, additional clonal aberrations occur, and the child then develops myeloleukemia of Down syndrome. So the very specific pathogenesis without any counterpart in adult hematology. My second example are the rhizopathies, which are clinically overlapping dominant disorders with cancer susceptibility. And as hematologists, we are particularly interested in Noonan syndrome. About half the cases of Noonan syndrome are coded by mutations in PTPN11, which is the gene for SHP2. Noonan syndrome is quite frequent with an incidence of 1 to 1,000. It's a developmental disorder, often with typical facial features, cardiac anomalies, most often pulmonic stenosis, but there also may be chest deformities, cryptorchism, short stature, and some children have severe failure to thrive. Now, the hematologist knows that there's a Noonan syndrome-associated myeloproliferation, which is a polyclonal disease and generally spontaneously resolves. There are very rare cases with monosomy 7. Now, Noonan syndrome-associated myeloproliferative disorder can clinically present like JMML. And juvenile myelomonocytic leukemia, this unique MDS-MPD overlap disorder, is my third example. It's defined by 5 canonical RAS pathway mutations. 2 of these mutations in NF1 and CPL are germline mutations with biallelic inactivation, and 3 of those are somatic heterozygous gain-of-function mutations in KRAS, NRAS, and PTPN11. The KRAS and NRAS mutations are the typical oncogenic mutations in codon 12, 13, and 61. Depicted here is also the frequency, and you see that PTPN11 mutated JMML is the most common subtype. Now, prognostic factors for survival in JMML have clinically been defined decades ago, and these are age above 2 years, hemoglobin F above 15%, and more recently, presence of subclonal mutations and DNA hypermethylation. Depicted here is more recent work indicating that these RAS pathway mutations define epigenetic subclasses, and as we'll show in a second, they also have prognostic impact. And you can see here that on the left side in red, in the hypermethylated group, many patients with PTPN11 are located, but also in the intermediate methylation group and some patients in the low methylation group. You can see that NF1 mutated JMML basically is in all 3 methylation groups, whereas KRAS mutated JMML kind of clusters in the intermediate methylation. And interestingly, many of these patients have monosomy 7. Also interestingly, NRAS mutated JMML is in the low methylation class, as is CPL, and as you would expect, Noonan-associated myeloproliferative disease, which I do not count within the JMMLs. So that transfers into overall survival according to methylation group, with the high methylation group, patients with a very high relapse rate of 40 to 50%, as opposed to the low methylation group with an overall survival of here 87%. Now, interestingly, the high hemoglobin F in JMML is regulated epigenetically. And in other words, our methylation pattern here, analyzed by whole genome methylation, very nicely reflects the hemoglobin F as well. And I could show you very similar pictures just with the hemoglobin F. So the message is many patients with PTPN11 mutated JMML belong to a high methylation group, have high hemoglobin F, often older, and a high relapse rate, whereas patients with KRAS mutated JMML are within the intermediate group, and patients with NRAS mutated JMML often have a low hemoglobin F, are in the low methylation group. So that's a very unique biology as well in an MDS-MPS overlap syndrome without any counterpart in the adult world. Now, the first clues to MDS pathobiology in pediatrics really came from the inherited disorders of bone marrow failure, with the increased risk for myeloid neoplasia. And I try to depict here the sequence of events from birth with a normal number of stem cells, then to aplasia with increasing bone marrow failure, the first events of clonal hematopoiesis, and then further events leading to overt MDS, and then progressing into MDS with excess blast or AML. Now it's of interest that these first acquired somatic events are in fact often driven by somatic compensation. And I put down two examples here. One is Fanconi anemia. And at the last ASH meeting, our colleagues from Paris with Dr. Soulier very nicely showed that there are unbalanced chromosomal translocation, or that the unbalanced chromosomal translocation 1p+, which has been known for a long time, leads to an increase of the expression of MDM4, which then downregulates p53. Another example comes from the work of Stockman-Diamond syndrome of Akiko Shimamura and her group. She showed that there is EF6 haploinsufficiency by point mutations or deletion of 20q, which in fact improve ribosome function. And then there can be somatic transformation as a result of a maladaptive compensation. And again, I take the Stockman-Diamond syndrome, where monoallelic p53 mutations can be adaptive, but then biallelic p53 mutations occur, which are clearly oncogenic. Traditionally, there was a separation of primary versus secondary pediatric MDS. And we had defined secondary MDS as MDS after a pre-existing problem, either therapy-related or in acquired aplastic anemia or in the classical inherited bone marrow failure syndromes. All other MDS were traditionally called in pediatrics as primary MDS, and the reason behind that was they manifest as primary disease, although it was clear that it's reasonable to assume that most of these disorders, in fact, are caused by yet unknown genetic predisposition. More recently, we identified some of these genetic predispositions, and in pediatrics, GATA2 mutation account for about 7% of all so-called primary MDS, and 79L syndrome of all, about 8% of so-called primary MDS, and the other genetic predisposition disorders like RUNX1, germline disease, for less than 1%. So in total, about 15 to 20% of all primary MDS is explained by germline predisposition. Most of these cases present as refractory cytopenia of childhood. And I bring this histopathology here because it is very well defined, and in the update of the classification and the ICC classification, we developed it even further. 80% of cases with this refractory cytopenia with a normal blast count are hypercellular, and you can see here in the AG staining and fluorescein esterase staining, patchy hematopoiesis with reduced granulopoiesis. Erythropoiesis is left-shifted. Increased numbers of erythroblasts and megaloblastic changes can be noted. And on the right side in the AG staining, you see this nest of left-shifted erythropoiesis. There's a marked decrease in megakaryopoiesis, often micromegakaryocytes with separated around nuclei, and you have to do immunohistochemistry to evaluate megakaryopoiesis and micromegakaryocytes. Now, the histopathological pattern with persistent cytopenia and the dysplastic changes in the erythropoiesis with the clusters of more than 20 erythroid precursors, the dysplastic granulopoiesis, which is reduced, and the megakaryopoiesis is being described in the last WHO. And as I said, we defined it even further to have pathologists around the world recognize this pattern. About close to 70% of these children have normal metaphase cytogenetics, but monosomy 7, other karyotypes or even complex karyotypes can be present. And in patients where metaphase cytogenetics do not grow, we can do FISH for monosomy 7 and trisomy 8. And no result means here that they are absent monosomy 7 and trisomy 8. Now, what can this histopathological pattern of refractory cytopenia in fact do for us? I think it separates an intrinsic bone marrow disease from reactive changes. It also separates refractory cytopenia from severe aplastic anemia at the time of diagnosis. What can it not do? It cannot distinguish clonal from non-clonal disorder. And we have to acknowledge that refractory cytopenia pattern is also present in some classical inherited bone marrow failure disorders, like, for instance, Fanconi anemia. And it's the same pattern we find in the newly discovered dominant predisposition disorders. So RCC, in fact, represents a biological spectrum of disease stages reflecting the plasticity of the bone marrow in children. It also helped us to develop an algorithm for management of these patients with refractory cytopenia. There we exclude the classical inherited bone marrow failure. And this algorithm has been highly successful in clinical practice. That's the algorithm our European working group currently uses for treatment of patients with refractory cytopenia, and it's based on the karyotype and the cellularity. Obviously, patients with refractory cytopenia and monosomy 7, 7q, or 2 or more operations are candidates for early hematopoietic stem cell transplantation. And you can see here, we currently in Europe use a conditioning regimen of tiosulfan, thiotepa, and fludarabine. On the left side, you appreciate that there are patients who are not transfusion dependent, who have an ANC above 1,000 per microliter, who are candidates for a watch and wait strategy. And in the middle, you see depicted patients with transfusion dependency, but not monosomy 7 or one of the adverse karyotypes, who are subjected to stem cell transplantation, and depending on the cellularity in the bone marrow, we choose the conditioning regimen. There's a small group of patients with very empty bone marrow, often patients where we don't see any megakaryocytes, who do respond to immunosuppressive therapy. But this is a very selective group, and there is no study comparing stem cell transplantation to immunosuppressive therapy. It's more at the discretion of the local physician. With this kind of algorithm, we actually did quite well, because I show you here data from our most recent paper on 79L, where we looked at the overall survival in refractory cytopenia from diagnosis. And you see on the top for patients with normal karyotype, top left and top right with monosomy 7, deletion 7q. And you appreciate that there is no difference between patients with 79L mutation, GATA2 mutation, or wild type. The bottom shows you the same data with the overall survival from the time of hematopoietic stem cell transplantation. And I think that's quite important for us, because the thinking here is that we can move the field together for patients with refractory cytopenia. There's currently no need to come up with specific conditioning regimens for stem cell transplantation in our hands for GATA2 or for 79 or 79L in RCC. The same is true for MDS-EB, but I have to note that we transplant these patients without intensive chemotherapy prior to stem cell transplantation, prior to the procedure. So now depicted here, the germline predispositions to myeloid neoplasia, which we may see in pediatrics or adult MDS. And I pointed the two GATA2 deficiency and 79L in red, because these two are primarily seen in children. We do see some patients with RUNX1 deficiency, very rarely ETV6 mutations. DDX41 is almost unheard of in pediatrics, and ANKD26 or ERCC6L2, again, are rare occurrences in children. I also put down the reported karyotypes, and you see the importance of monosomy 7 and the congenital abnormalities. Now, a few words for GATA2 deficiency. It's a phenotype defined by immunodeficiency and constitutional abnormalities. And in this slide, you see the frequency of autism or ADHD, congenital deafness, quite frequently kidney abnormalities, warts, lymphedema, or immunodeficiency. And about 51% of patients have any abnormality. The mutations we find in GATA2 are either truncating mutations prior or within the second zinc finger, or missense mutations within the second zinc finger, but we also see non-coding mutations in exon 4. We do see gene deletions and silent mutations. This indicates that it's not sufficient just to do a somatic panel. If you expect GATA2 deficiency, I think you particularly have to analyze and search for these alterations. One still very puzzling feature are the different hematological phenotypes in the different age groups. And I bring you one example here. I once took care of a patient which was 16 when I saw her first with a hypoplastic refractory cytopenia and a trisomy 8. She did not want to be transplanted. She went on to see adult hematologists. They put her on EPO. In fact, she lost her trisomy 8, but she represented at the age of 31 with AML, 80% blast, trisomy 8, and an NPM1 mutation. We became aware of that because at the very same time, her daughter presented with MDSCB, monosomy 7, and a somatic ACLX1 mutation. They both carry the same GATA2 mutation in zinc finger 2. So the question is, why do in pediatrics we see more often this sporadic MDS type with monosomy 7 without preceding phenotype and GATA2 deficiency, whereas our adult colleagues see more often the phenotype with cellular deficiency and bone marrow failure? I think it's still unanswered. And well, we'll see how we can solve this puzzle. I think it's important because if we see adolescents with trisomy 8, it becomes very hard to counsel them for the right strategy. If we would have transplanted the 16-year-old with trisomy 8, she may not have become a mother at the same time. Unfortunately, she succumbed to her stem cell transplantation while her daughter survived. Now, if you just look at monosomy 7, GATA2 deficiency accounts for about 30% of all monosomy 7 in pediatrics, and there is a clear age dependency. If you see an adolescent with MDS and monosomy 7, his or her likelihood as having a GATA2 deficiency is about 70%. Now, the other syndrome, 79 and 79L-like disease, is even more exciting in my mind, because 79 and 79L are bona fide tumor suppressors. They are paralog genes on 7q with a high sequence identity. The function is not clear, but they terminate signaling downstream of growth factors. They're anti-proliferative. They're involved in interferon response and antiviral immunity. And knockout mice developed myeloid malignancy. There is a disorder with delirious mutation called normophosphatemic familial tumoral calcinosis, and you appreciate here the tumors from calcinosis. Now, germline disorders with gain-of-function mutation in 79 and 79L have been described recently, and they share a hematological phenotype. One is Niemeyer syndrome, germline mutations in 79, with adrenal hypoplasia, genital phenotypes, and growth restriction. And the other one is ataxia pancytopenia syndrome with cerebellar and hippocampal hypoplasia, neurodegeneration, and other malformations. Both can show hematopoietic failure with the risk of MDS. And interestingly, chromosomal 7 aberrations are frequent and can be transient in small children. Now, the gene 79 or 79L are located on 7q21. And the mutations we find are missense germline mutations with a gain of function, resulting in severe inhibition of proliferation and survival of the hematopoietic stem and progenitor cells. So there are multiple rescue mechanisms of these stem and progenitor cells, like development of monosomy 7, like deletion 7q, like uniparental disomy, which is doubling of the normal allele here, and they all result in a loss of the mutated gene, but there also are acquired protein truncating mutations with a loss of function. Now, uniparental disomy obviously is a genetic correction. Acquired protein truncating mutations can result in partial compensation, but monosomy 7 and deletion 7q are oncogenic events. And in the bone marrow of these children, you see all these different clones running against each other. And the question is, which clone will take over? One example here, and you see the normal 26XY clone in black. You see the monosomy 7 clone in red, and you see a deletion 7q clone in blue. Our current practice in small children below the age of 5 is to follow these patients with 79 and 79L syndrome closely, follow their clones in the bone marrow, because some of the children will in fact experience UPD and cure themselves. However, the majority of these children in our hands need bone marrow transplantation. They can be severely pancytopenic. They also have an immunosufficiency, and they certainly should not be treated with growth factors, but rather transplanted, because I showed you that the event-free survival of the children we transplanted is in the order of 90%. If there's a normal karyotype and monosomy 7, it's around 70 to 80%. We systematically analyzed a huge cohort of children with MDS for presence of 79 and 79L syndrome, and published that last year. And we also had some selected cases to describe the constitutional phenotype, the variant evaluation, and the clonal events. And I'll show you some pictures from this Nature Medicine paper. This is the picture showing 79 mutations on the top, 79L mutations at the bottom in the 79 gene, and they cluster into what we now call middle region. You see cases with normal karyotype and cases with monosomy 7. And on the in the blue print here, you see the somatic rescue mutations. I must say it's still very difficult to allocate these variants we find, and we have to take into account the allelic frequency in the different populations, because there is no really good functional test. Somatic rescue mutations are in fact very frequent, and Martin Brodarski and his lab, who worked on these studies, looked at a lot of single-cell DNA sequencing analysis and you can see that they found acquired events in about 64% of all 79 and 79L patients. Within this cohort, the constitutional phenotypes of mirage and ataxia telangiectasia were somewhat blurred. In a high percentage of patients, we find these endocrine, psychomotor, neurological, skeletal, gastrointestinal, etc. abnormalities, but it's kind of hard to really pinpoint the gene. There is a very broad overlap. Now, with GATA2 and 79, 79 in blue here, GATA2 in orange, we unraveled, as I already said, about 15% of all pediatric MDS as germline disease with one of these two genes involved. It's also 15% in refractory cytopenia of childhood. And if you look at MDS-EB or MDS-EBT, it's about 21%. Just looking at the cases of monosomy 7, we unraveled about half of all monosomy 7 pediatric MDS cases. But the distribution between refractory cytopenia and MDS is different. More cases in MDS-EB have a GATA2 mutation than 79L mutation. But you also see that for the normal karyotype, there is still a lot to discover, and only about 9% of all patients with normal karyotype have one of these two mutations. Now, the mutational landscape in children has been unraveled by a number of excellent research groups, and I'll show you some data from Jason Schwartz from St. Jude. They looked at primary MDS, which was half MDS-EB and refractory cytopenia, some cases of MDS-MPN overlap syndrome, and some cases of AML with multilineage dysplasia. And what they found is that spliceosome mutations, which you find in adults, are almost virtually absent. In fact, you see most commonly RAS, MAP kinase mutation, CoAC mutation, some epigenetic modifiers, some transcription factors, and some gene processing or RNG genes. So the landscape is quite distinct from what you know from adult MDS. Now, when I think about MDS, I think about MDS as the disorder squeezed in between myeloproliferative neoplasia, AML, refractory, and bone marrow failure. It's really a squeeze-in disorder. And it's kind of interesting that Dr. Hassayan just pointed this out in his educational book in 2021, because he said classification systems optimally identify diseases that retain the core ontogeny of the original presentation. And that's my problem when I think about MDS and AML, because we use the term AML on the one side for a biologic entity, and on the other side for an arbitrary blast cutoff of 20 or formal 30%. The separation of MDS from AML is, however, crucial for young individuals. And I think you all agree with me that this patient, number one, 10 years old, with 23% blast, M2 morphology, orbital chloroma, translocation A21, and rapidly progressing disease, has AML, and most likely can be cured by intensive AML therapy. And at the same time, the other patient, number 2, with 16% blast and a hypercellular bone marrow dysplasia, monosomy 7, and a germline GATA2 mutation, has MDS-EB and GATA2 deficiency, and has a good chance of being cured by hematopoietic stem cell transplantation. And as I said, because it's also an immunodeficiency, we are quite careful in using intensive AML therapy prior to transplantation. So the separation of MDS from AML being important in pediatrics may not matter that much for MDS in older adults. And on the left side, you see some sentences from a paper from Seth Curry, when he said that the prognosis in adults with MDS or AML is dismal and the prognosis grim. And data from Dr. Papagnoli on the right side, looking at different number of driver mutations, really shows that. So I understand my adult colleagues when they say we have to really tackle this terrible prognosis for our adults with MDS or AML. We want to put them into clinical studies. So he says defining patients with 10 to 30% blast as AML MDS eligible for AML or MDS studies would allow patients access to more therapy and potentially simplify the regulatory approval process, meaning here the FDA. This may be appropriate for adults with MDS, but I think, as I tried to point out, for pediatric MDS, it's really crucial to see whether it's MDS or AML. Even some cases with MDS and a blast percentage of 10 to 30% may behave more like MDS than AML, and that's the old RAP-T. And in pediatrics, we had the first problem when the 2001 WHO classification came up, and Hanny Castle pointed out that there is a small group of MDS EBT, which biologically should be included into MDS strategy. And even the 2016 WHO classification leaves us a little hole, although we stick to AML with 20% blast as a cutoff, to include these patients in MDS strategies. So in summary, MDS in pediatric patients is quite different from MDS in adults. It is less common. We do not see certain subtypes like subtypes with refractory cytopenia. We have probably, and this is a hint, if you include the MDS after the congenital bone marrow failures, 30% or more who are associated with germline disease. The 5% in adults over the age of 20 may be underestimated if I account for the DDX41 cases, which are now come up. We have more patients with familial disease and the chromosomal aberration, as the molecular alterations which we see as germline or somatic mutation are quite different. And I think I pointed out that spliceosome mutations are really very frequent, and more importantly, the aim in pediatrics is curative, and unfortunately, in many of the adult patients, only palliative. And with this, I would like to acknowledge our European Working Group of MDS in childhood in 20 countries with the regional coordinators, the reference lab, the transplant teams, but also my own collaborators here in Freiburg, and specifically Martin Brodowski, who some years ago, 3 or 4 years ago, moved from Freiburg to St. Jude and did these wonderful studies on 79L together with Sushi Saho. And obviously our international collaborators are Kiko, Shimamura, David Williams, Mitch Weiss, and Jerome Soulier. And I would like to thank you for your attention. Being the last speaker of the session, as I heard, I hope I still could tell you some new ideas or bring you up to date of what pediatrics thinks about MDS. Thank you very much indeed.
Thank you, Dr. Charlotte, for this wonderful update. We enjoyed listening to you. Every time we listen to you, there is a lot of information to be learned. So we have some colleagues over here who would like to discuss certain things with you. In the question box, we have a question from Dr. Raj. How can we predict which of the newborns with TMPD and GATA1 will go on to develop leukemia? Any clues to who would receive aggressive treatment?
Yeah, I think that's a very good question. And maybe I can expand there. There is this, I always say, incubator refractory cytopenia, which is a certain pattern. And within this pattern, within this incubator, you have oncogenic events, you have genetic reversion. I showed you in 79, it goes back and forth in both directions. And I think you have to judge the events you see on the either germline predisposition or in the total picture. So for GATA2, we know that monosomy 7 is bad, and these children need to be transplanted. We also know that they have ZPP1 mutations coming up first. Now we find some RUNX mutation. We don't know what they mean. So I think it's important to register the patient and get the experience to know what these mutations mean. Like we did in Swartman, which I showed you the work, didn't show you in detail, like we know from Fanconi anemia. We have to understand the mutational landscape for each of these separate entities. That's my not really satisfying long answer.
Dr. Rajat from Singapore.
Professor Niemeyer, fantastic talk. Let me just take this opportunity to thank you. Even in a country like Singapore, we have sent you quite a few samples for the AMML, and with your incredible help in opinion, it has been really, really helpful for us. So thank you for that. I've got two comments to make. Actually, we do have a Mirage syndrome kid now with a monosomy 7 we are transplanting, which will probably just change hematological manifestation, but not the other ones, the intestinal and the respiratory problems. But it's good to see that you showed that the outcomes are pretty good. But the question I have is, I have a kid who's following with thrombocytopenia. She's just about 3. Mild thrombocytopenia, and just told today it's a RUNX1 deletion, which is significant. So do I have to do a marrow to dysplasia and other things, or can I just follow this kid? Just the incidental thrombocytopenia and nothing else. I'm listening. Thank you.
I think that's an excellent question, and these are questions nobody has the right answer to. And even the RUNX1 Foundation, where we collect all the cases, don't have enough data really to answer this question. We are just trying to come up with a consensus of the European Hematology Association and EVOC-MDS to give some expert opinion. And my expert opinion for that particular kid is obviously look in the family, and if there's somebody who had very early life, MDS or AML, you are more cautious. But at the same time, I think what we would do is do regular CBCs on this child. And if the CBC changes, then do a bone marrow. We had the experience when we started our work about 20 years ago. We proposed for all of these RCC patients to do a bone marrow every year. And what happens is they do it in the first year because mom brings them in. And when there are adolescents, they take off. They had enough. They never show up again. And I don't think we serve these patients. So my feeling would be to do a marrow initially, including all genetic studies, because if you just do a marrow, you're not fair. You then really have to look for all somatic clones or even rescue events. And then followed by CBC, and then you can argue to do a bone marrow every 2 or 3 years or something, but that already is a lot.
Thank you, Rajat. Dr. Sandeep.
Thank you, Professor. It was an excellent lecture. I have 2 questions. One, you mentioned that interesting phenomena of somatic adaptations, especially in SAMD9 mutations. So, how long do we wait for this? And you also mentioned that transplant outcomes are so nice. So how long do we wait for this, or do we just transplant as early as possible when there's donor available? And secondly, can we exploit these somatic adaptations as a gene therapy to treat MDS, maybe even in adults?
Yeah. First of all, I think there are more somatic adaptations or somatic rescue events, probably as we appreciate it. We find them in other disorders as well. So not every mutation is bad. Maybe some of them are really adaptive events. For the 79 and 79L, if you look in the literature, my reading is that these transient monosomy 7 occurs in small children, and we made an artificial cutoff for 5 years. So for EVOC-MDS, we currently follow these patients carefully, but I say carefully because we already lost children, which I think we should have transplanted earlier because of pancytopenia, because of immunodeficiency, which they have too. So if they are stable and you do regular marrows, and we do regular marrows on them every 3 to 4 months, and then look for the rescue clones, you can do that. But when in doubt, transplant. And if they're older than 5 years, we do transplant. The results are not bad. So we don't want to get overexcited because of this natural gene therapy and lose our patients. I believe, yes, it exists, but it's not that common that you really have complete correction. And even with these truncating mutations, you don't know what's going to happen in the end. So be careful. Under 5 years, if they are stable, endow transplant. That's the message we give.
Thank you. Thank you.
Thank you, Sandeep, for that question. Shilpa?
Thanks for the wonderful talk. I was just keen to understand this, that in childhood MDS with inherited bone marrow failure, do you actually, in the absence of an unrelated donor, especially in countries like India where you do not find a full matched unrelated donor and there is a financial constraints and you have you do not have a matched sibling donor and you have a haploidentical, do you push for a haploidentical upfront, or given the nature of the disease, do we consider haploidentical? And if so, do you change your conditioning regime? Because obviously the PT side comes and then you have a background of inherited bone marrow failure.
Right. You have much more experience than we do in haploidentical bone marrow transplants. So you are the expert. We are not. We only use it if we don't get an unrelated donor. So it's not on me to discuss haploidentical transplant. And number one. Number two, the conditioning regimen really depends on your way of doing haploidentical transplant. So again, I have to point back to you and your studies. All I can say, and that's important, if we look at our data on haploidentical transplant in JMML, they're terrible. But they're not terrible because haploidentical transplants are terrible. They're terrible because we are transplanting terrible disease. So I think to answer your question, if you don't have an unrelated donor in JMML or GATA2 deficiency, I think it is very reasonable to think about that along the same lines than our indication for unrelated donor transplants. It's not the last thing to do. It's, you know, it's the same indication. The kid will get, I didn't show you that, for GATA2 deficiency at age 40, 80% have AML or MDS. So it needs to be transplanted. But I think there, your transplanters are the expert, not we. Not very helpful, but I can't see any reason why well-done haploidentical transplant doesn't have the same importance than unrelated donor transplants. That would be my answer. Pointing back to you.
Thank you, Shilpa. Tathagat?
Good evening. A very nice presentation. Good evening. Nice presentation, ma'am. My question as a morphologist, I'm essentially a hematopathologist, and it's a nightmare to diagnose pediatric myelodysplastic syndrome on the bone marrow biopsy, despite all the repertoires of the immunohistochemistry which is now available with all of us. My question to you is, how often do you see myelodysplastic syndrome with fibrosis and its correlation with GATA2 insufficiency?
First of all, welcome, hematopathologist. There are not too many who look at pediatric bone marrows, but you probably see more. You have a different population than we have. Many hematopathologists probably share your opinion. That's why we worked on the RCC classification again, because we believe it's an intrinsic pattern. Myelofibrosis is not very common. If we see it, we most often see it with excess blasts, but we don't see that very common in our germline disorders, whether it's GATA2 or something else. Yeah. So uncommon is the answer, and mostly as an expression of the disease with increased blast.
Fair enough. And if I may ask a second question pertaining to that, why do we exclude refractory cytopenia with multilineage dysplasias in the diagnosis of RCC?
Well, we don't exclude that. You probably saw the leukemia paper and you probably saw the blood paper and you're totally confused. Yeah.
That's the point.
So that's the point. So I think to expand on that, we actually tried to be more stringent with the criteria for RCC. Because it's really a unique pattern, and we came up with this MDS loss, otherwise specified for all the other cases, to have a pot for them as well. And we strongly believe that this actually is the way to go. When we started off, we claimed all RCC is MDS, which it's not, because some of that is just not clonal. And it's this huge incubator with all these clones coming up and going down. The reason to drill on RCC is that, number one, these patients get a diagnosis rather than saying don't know, some unspecified bone marrow failure. And that means they're not lost and they will get a certain surveillance and we have a management for them. And that's actually my criticism. We're just writing a letter to leukemia on the current WHO classification. Which just says, okay, forget RCC, MDS is clonal, and we define childhood MDS without blasts, exclude all germline disease. There I have to ask, what the heck are you calling these disorders? Yeah, and it makes it very difficult for pathologists. Let's say 79, then a patient moves between MDS and no diagnosis, I don't have a name for it, and back to MDS. And that's difficult. So rather acknowledge that RCC is a little bit like seekers, these strange disorders where you don't really know what they are. Yeah? That we have that and that there is plasticity and that we don't have better means. We only look at genetic changes. We have not looked at epigenetic changes. Yeah? We don't understand most of that. But at least I find some pattern and I call this pattern RCC. I can follow these kids. And some of this RCC probably is bona fide MDS. Some of this is not. But you can decide what you call MDS and what not, because depending on the change you see, this may be a rescue event or this may be an oncogenic event. It's just complicated. You know?
I agree. Thank you so much. I agree. It is confusing, but you have cleared it out. That's important. You have cleared it out. Thank you so much.
Yeah, thank you.
Thank you, Dr. Charlotte. So that was the last question. We're grateful to you for sparing your time. It's lovely. Each time it's great to for the official one. Thanks, we have Dr.
Yeah. Thank you very much for having me.
Thank you.
Thank you. Thank you, Professor Charlotte. You have to say good morning, good evening, and good night to a lot of people. And reason I'll tell you just now. Reason is we have participants from India, Abu Dhabi, Iraq, Malaysia, Africa, Sri Lanka, Bangladesh, Australia, Myanmar, Singapore, Tunisia, Egypt, Indonesia, Pakistan, USA, Ethiopia, and UK.
Well, we are absolutely global. Thank you very much for that opportunity. Thank you.
Thank you so very much. And a big thank you also to Dr. Daniel Hodson and Professor Samira for other two lectures. Thank you to all the expert discussants, Dr. Bharat Patodia, Dr. Hari Menon, Dr. Rajat, Dr. Kunga, Dr. Verma, Dr. Sampath, Dr. Saurabh Bhave, Dr. Kunjan, Dr. Tejaswini, Dr. Jyoti, Dr. Radhika, Dr. Dipankar, Dr. Shruti, Dr. Kunal, Dr. Rashmi, Dr. Prashant, Dr. Avnish, Dr. Shyam Agarwal, Dr. Girish, Dr. Samir Melankari, Dr. Rahul Arora, Dr. Krishna Ratnam, Dr. Rajkumar, Brigadier Tathagat, Dr. Sanjeev Gupta, Dr. Datta, and Dr. Sandeep for those interesting questions throughout last 3 hours. And last but not the least, to Professor Dr. M.B. Agrawal, sir, for pulling off the blinder. I just wanted to give one more reminder tomorrow 6:25 till 9:30, we have the presence of Dr. Beverly from London and Dr. Beatrice Lecter from Switzerland and Dr. Linda Larsen from Sweden. So please log in again. Thank you so very much, and all of you have a wonderful evening and good night.
Thank you. Bye-bye.